Je suis une danse, lente et majestueuse. Une boule disco qui scintille de mille feux, triste et seule dans une boîte de nuit, tournant au rythme des verres qui se vident et se remplissent. Je suis ton absence qui devient ma seule présence. Je suis l'espoir d'une fille, impétueuse et fatiguée du monde fou qui l'entoure. Je suis mes amours, mes peurs et mes chagrins. Je suis l'attente en mouvement. Un roman d'amour qui n'a pas encore été écrit. Je suis cette robe rose qui me fait danser dans les bars, légère et courte, comme notre histoire. Je suis tes yeux bleus désormais lointains, et les prochains. Je suis la nuit en cacophonie, qui étouffe mes larmes pour les rendre plus jolies. Le bruit de mes pas marchant sur le trottoir mouillé dans la nuit noire, et le silence, assourdissant d'avoir trop espéré. Qui n'a pas encore été écrit.
Now it's me and you. Now I've been waiting, think I wanna make the move. Now, baby, tell me if you like it. Tell me if you like it. Now I've been waiting, think I wanna make the move. Now, baby, tell me how you like it. As long as you're cool, right here is where you wanna be. Baby, tell me, baby, baby, tell me. Right here is where you wanna be. Baby, tell me, baby, baby, tell me, to me. Now it's me and you. Now it's me and you. Now think I wanna make a move. Now as long as you're cool, right here is where you wanna be. As long as you're cool, baby, tell me how you like it. As long as you're cool, here is where you wanna be. As long as you're cool, baby, tell me how you like it. Oh yeah, yeah. As long as you're cool, right here is where you wanna be. As long as you're cool, baby, tell me how you like it. As long as you're cool, here is where you wanna be. As long as you're cool, baby, tell me how you like it. Oh yeah, yeah. Oh yeah, yeah.
There's something about you. 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 What about a hit? What about a hit of your love? Start to shake with your hand. Will it ever click? Will it ever click? Are you a freak? You turn and face me. Maybe this time I'll choose. What about a hit? What about a hit of your love? Start to shake with your hand. Will it ever click? Will it ever click? Are you a freak? You turn and face me. Maybe this time I'll choose. What about a hit of your love? Start to shake with your hand. Will it ever click? Are you a freak? Maybe this time I'll choose. What about a hit of your love? Start to shake with your hand. Will it ever click? Are you a freak? You turn and face me. Maybe this time I'll choose. Closer, closer, closer. You turn and face me. Maybe this time I'll choose. Closer. Closer. You turn and face me. Maybe this time I'll choose. Closer. Closer. You turn and face me. something about you. There's 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 something about you. What about a hit? What about a hit? Oh, the start to shake. Start to shake with your hand. Whatever click. Whatever click. Are you a freak? You turn and face me. Maybe this time I'll choose. What about a hit? What about a hit? Oh, the start to shake. Start to shake with your hand. Whatever click. Whatever click. Are you a freak? You turn and face me. Maybe this time I'll choose.
Tell me that you will never let go. Voice like an echo. You will never let go. Voice like an echo. I can hear your voice like an echo. Tell me that you will never let go. Voice like an echo. You will never let go. Voice like an echo. I can hear your voice like an echo. Saw you dancing by the ocean, running fast along the sand. A spirit born of earth and water, fire flying from your hands. Hold me closer, time to dance. Count the headlights on the highway. Lay me down in sheets of linen. You had a beautiful day today. Hold me closer. Hold me closer. Hold me closer. could be something me and you. When you call my name, it's like I'm falling apart, like I'm striking my heart. Your head on my face, can feel myself drifting off into another place. Unconsciously I think I'm falling. Maybe it's just something I do. I feel like we could lead to something. Maybe we could be something I do. It's like I'm falling apart, like I'm striking my heart. It's like I'm falling apart, like I'm striking my heart. Look at me as we're getting older. Finding out that it's something people do. Seems like by now we should be over, but I'm not ever getting over you. When you call my name, it's like I'm falling apart, like I'm striking my heart. Your head on my face, can feel myself drifting off into another place. Unconsciously I think I'm falling. Maybe it's just something I do. I feel like we could lead to something. Maybe we could be something I do. It's like I'm falling apart, like I'm striking my heart. It's like I'm falling like I'm striking my heart. When you come alive, when you try to chase it, when you feel you want it, when you feel the weight, when you wanna lie, but you don't wanna fake it. Our eyes become one, maybe it's fate. Can you feel alive? When we should try to chase it, when you wanna walk, when it's getting late, when we need a call. You're the story, every night. You're the story, every night. When we feel alive, baby, don't go away. Try to make it back home. When we feel alive, baby, don't go away. Try to make it back home. When you come alive, when you try to chase it. When you come alive, when you try to chase it. When you feel you want it, when you feel the weight, when you wanna lie, but you don't wanna fake it. Our eyes become one, maybe it's fate. Can you feel alive? When we should try to chase it. When you wanna walk, when it's getting late, when we need a call. You're the story, every night. You're the story, every night. You've been so good to me. Your love inside me. Oh, baby. You're my consolation. Oh, baby. There's no hesitation. Oh, baby. You're my consolation. Oh, baby. There's no hesitation. Every sky will be blue, long as you're loving me, loving me. You've been so good to me. Give you anything. You've been so good to me. Your love inside me. Oh, baby. You're my consolation. Oh, baby. There's no hesitation. Every sky will be blue, long as you're loving me. You were right. I know I can't get enough of you. Leave it all to bloom. You were right. I know I can't get enough of you. No, the things that I would do under the water. I'm sinking further down, floating on nothing. I'm nothing. Arms are wide open. I'm calling out your name. Sitting and waiting. I'm waiting. You were right. I know I can't get enough of you. You were right. I know I can't get enough of you. Leave it all to bloom. You were right. I know I can't get enough of you. No, things that I would do. Cold in the river, lips moving, there's no sound. Pulling me under, I'm under. You're giving me nothing. You keep telling me I'll be found. Keeping me guessing, I'm guessing. You were right. You were right. I know I can't get enough of you.
gets me high. I can live in this moment for the rest of time. So stay a little longer, my love. I just can't get enough of all these summer skies. They wash away the darkness from my mind. I can live in this moment for the rest of time. Oh my, I just wanna dive into your love, your love. Dive into your love, your love. I just wanna dive into your love, your love. Dive into your love, your love. I just wanna dive into your love. I just wanna dive into your love. I just wanna dive into your love. I feel there's something coming over me. It's like a flash in my head. Right here with you is where I'm supposed to be. It's like coming home again. I just can't get enough of all these summer skies. The sun, it brings me up, it gets me high. I can live in this moment for the rest of time. So stay a little longer, my love. I just can't get enough of all these summer skies. They wash away the darkness from my mind. I can live in this moment for the rest of time. Oh my, I just wanna dive into your love, your love. Dive into your love, your love. I just wanna dive into your love, your love. Dive into your love, your love. I just wanna dive into your love. I just wanna dive into your love. I just wanna dive into your love.
All right. Good morning, everyone, and welcome to our symposium on artificial intelligence in structural biology. This is the second time we organize this event, and I'm very happy to see so many familiar faces here today. Also, a warm welcome to the people who joined online, as we are also streaming this session live. The event is organized by the Flemish Supercomputer Center, VSC, together with EuroCC Belgium, the two centers that represent high-performance computing, respectively, in Flanders and in Belgium. A couple of years ago, AlphaFold was one of the most requested software packages ever on our supercomputers. And with its incredible development speed and impact, it was clear to us that we need to host a community on this topic. The past couple of years, this community has hosted a series of activities, such as trainings and webinars. And through VSC and EuroCC Belgium, we also offer support to researchers and to companies on these topics. We are grateful for your participation and interest, and we'd like to thank the FWO, Vito, and VIB for their support in the organization of today's event. I would also like to thank the three speakers here today, Arne Elofsson, Casper Goverde, and Maxim Tsenkov, for traveling to Brussels today to present their fascinating work. And now I would like to hand over to Shoshana Wodak, who is the chair of this morning's session. Thank you.
Okay, it's a pleasure for me to chair this session this morning and this important event. And I'm presenting the speakers. The first speaker is Arne Elofsson. Arne did his PhD at the Karolinska Institute in Sweden and has developed most of his career at the University of Stockholm and did a postdoc at the time with David Eisenberg. And his research focuses on developing methods for deep learning methods and their application to structural biology. And he aims at trying to provide a more complete structural coverage of the proteome. And we'll hear what he has to say about his work. Please.
Thank you. Let me see if we switch to my slides. So thanks for inviting me. It's a pleasure to be here, and I guess if someone has some questions, just ask. And online, I guess you have to write in some chat or something, and someone has to read it out because I can't see it. I thought I should give a little bit of background. I mean, this is a story that started some years before AlphaFold and continued at least 1 or 2 years after it. And it's a bit of a hint for the future also. But I will tell you a little bit what we have done, and well, not only we, but the field has done for the last years. But I'll start with a very short introduction of what a protein is. If this one works. No, yeah. So protein, as I'm sure most of you know, is a chain of amino acids. And already 50 years ago, it was shown that, or 70 years ago, it was proposed that it could form what's called secondary structures. So Linus Pauling modeled it and showed that you can form helices and sheets and some other structures also that are basically satisfying the hydrogen bonds of the backbone of the amino acid sequence. And then it was shown that it folds into a three-dimensional structure. And that has been shown that it's for most proteins, or at least for many proteins, this is this global free energy minimum. It's not always the case, but in general that's the assumption that most cases have that. And sometimes, as you can see here, it binds some kind, I think you can see mine, yes. You can see it binds some other molecules, in this case a heme group, so do some function. But then also, it also folds together with other proteins and form complexes. So this is hemoglobin, and it consists of 4 chains. So there are proteins consist of many chains, but there are so interaction between proteins is also important, not only proteins and things. But it's all governed by the same physical rules. So yeah, here we have this, and there were some traditional methods to do the structure prediction, and the only thing that really worked until recently, until basically 2021, is that you use homology. So basically the idea is that 2 proteins that have a common ancestor, that are homologous, actually in most cases have a very similar structures. So if you know the structure of one of them, you can use that to model the other one. And this applies both to single proteins, but also to multi-proteins. It's not always the case, things can change, but in general, that's the assumption. And many people spend years on improving these methods, and it's based on improving the alignment of 2 sequences or a sequence and a structure, but also using parts of sequences like fragments and bringing things together, etc. So there was a lot of things that worked at. So that works for a quite large set of all proteins, but it doesn't really explain how proteins fold. So there were also people for many years have tried to fold proteins in using some kind of physics-based models, more or less detailed, but basically I think people end up doing it that you need to at least have an all-atom molecular dynamics model, including water. And you can actually fold small, fast-folding proteins this way. You can take something that's short and folds in milliseconds, put it in a supercomputer, and run it for a long time, and get a good description of the free energy landscape. But as you can see here, it's often, if you don't have enough time, you can get stretched into local minima. It takes time, and there's a lot of physics to understand there. But that's, in theory, if the methods were good enough and we had fast enough computers, we could simulate proteins like that. But we are quite many orders of magnitude away from that yet. And it's not clear that the methods really are accurate enough to do that. So what has worked, what started working some 10 years ago is the idea is 30 years old, it's in the mid-90s. It's the idea that you can use evolutionary information because we assume in the evolution that the structure is conserved. So the structure will have constraints on evolution also. So that means that if you have two residues that are in contact, like that residue and that residue, they could have an evolutionary pattern that's sort of, if one changes, the other one should also change. So if you have a big amino acid and a small one, the big becomes small, the small one has to become big, so fill out the gap. So it should be co-evolutionary signals there. And this was a couple of groups that did it in the mid-90s. It did not really work. I mean, it had some value, but it was not good enough for really predicting structure. But people used it as filters and so on. And it was even almost an abandoned field because we had it as a category in Casper to predict contacts, but then basically it was abandoned because progress was so small. But then it's one thing that people realized, and it's actually realized in '99 by Lapidis. And there's also a couple of other papers about the same time, with the same idea. But that was sort of forgotten because it didn't really, well, it probably worked, but probably the data was not there and people didn't read the papers. But the idea is basically that if you have, think about a protein like this, you have a native protein, it's well packed, and amino acids are nice together, this is a small part of it. And then for some reason there's a mutation. Mutation happens spontaneously, and that will of course somehow disrupt this packing. So that will be unfavorable, but of course, if it's not too bad, the organism can still survive. But there's a tendency to have compensating mutations that are going to happen. I mean, if you have compensating mutations, they're going to be favorable. So they're going to be compensating mutation. So you first mutate the pentamer to an arrow, and then you mutate the quadrat to a pentamer, and then it looks nicely packed. However, if you look at the top here, look at this region here and this region here, they're not the same. So they're going to be indirect interactions. So you're going to have indirect couplings. And this means that if you have residue I interacting with B, with J, and J with X, then I and X are also going to have contact. And because all residues in the protein are connected, this indirect coupling is going to cause correlations that you don't pick up if you look at directly. But that was compensated for using, for instance, this direct coupling analysis. So you have indirect coupling, and you can compensate for that. And that really changed the area of contact predictions. So really, suddenly, contact predictions become useful. So this was Martin White and others who did this in around 2008 started. And suddenly, people could model the structure of proteins. And Debbie Marks and Chris Sanders and others pushed you can actually do it for quite a lot of proteins. However, so you do this list. You do something like this. You take a protein. You look at the contact map. So this is the sequence of the protein, the first, and this is the sequence. And whenever there is a gray dot here, there's a contact. So this is some contacts here, down here. And then this is a method that predicts it. And in this case, you predicted all the green ones, and you predicted red that are correct, and you predicted a few extra ones. But you really can see here that you can predict quite a lot. You miss the long one range information here, but you miss the short ones. You can predict the pack on these sheets together. However, you can also see that these contact patterns are clearly not random. It's not like a random distribution of Gaussian noise. There are patterns there. There are like diagonals. There are contacts that are close to each other because the structure has information about the contacts. And of course, patterns is something that machine learning is really good at using. So machine learning is good at recognizing patterns. So if we can see it, it would be quite easy to train contacts. You have to think the simple thing. If you have one contact here and one contact here, it's likely to have a diagonal along it. So you can look at things. You can easily see if you have a single contact at one point and nothing else around it, that's quite likely false positive. But or either that or you have more contacts around it because you don't have single contacts hanging around. So we did this in 2014. Was our first attempt. So this was our very naive deep learning methods for doing this. So we took 2 of these methods for predicting contacts, PLM-DCA and Tsenkov, that are, the same idea, but there are different mathematics behind them. And we also use 4 different multiple sequence alignments. And we had 8 different multiple sequence alignments. Basically, you can use multiple sequence alignments, the input here, you can use different cutoffs for the iteration, you can use different methods. We tried to figure out what was the best method, and we figure out, as many other cases, that there's not only a single answer to that question. It depends from case to case. In some cases, you get better patterns using one. In some other cases, you get better patterns using another one. So we took this, and we added some information about secondary structure and surface, etc. And we had a simple, in this case it was very simple. It was random forest to predict the contact. But then we iterated. We had a predict contact, and then we iterated here a number of times. And we showed that for each iteration, at least for 5 iterations, we could do better and better predictions. And so at the end, we could go from this noisy picture here on the top here to something that actually has a quite good prediction value here, down here. So from 18% to 56% correct context predicted. So in this case, at least it worked, but yeah, so we do that. And we showed then we improved this method, we used more advanced machine learning methods, and we showed that this actually could help quite a lot. So this is the overall performance of like many years of work together. Here, in the x-axis, we have the size of the multiple sequence alignment. The problem with these earlier methods is that you needed quite big multiple sequence alignment. You needed 1,000 sequences. And in 2014 or so on, that was not for all families you had that, because the sequence was not, nowadays, you basically have it for everything, because the sequence database is much bigger. But it was a problem. And then we had some measure here, this is just the number of correct predictions. If you do 1 prediction per residue, so you predict maybe half of the predicted content should be there. And you often needed something like 40–50% there to make a good model. It was not a straight cutoff, but at least it's a good measure to measure there. And if you look at this is the DCA type methods. They are, there's a few different ones, and they are, you see that if you get over 1,000 sequences, they're getting quite good. But for small families, they're getting quite bad. The red line here is an old method, much earlier, that is based on machine learning. It doesn't use these indirect couplings. So it never reaches a very, very high performance. It reaches a performance of maybe 30%, 20–30%. And that was state of the art, basically, before. But it's quite flat. It does it even for the small multiple sequence alignments. This is quite flat. For some reason, there are some hard ones here around the 1,000 that is more difficult. So everything has more problems there for some reason. And then we showed that we could, by machine learning, basically combining these two type of informations. It's not the same method, but we could basically improve it all over the target. So even for like things with 100 sequences, we could actually do good predictions. Still, if it was less than 100, the performance dropped quite rapidly. So that means that we could model things for most big protein families. So we developed a pipeline, called it PCONS-fold. So basically, we took these sequences, we run a number of multiple sequence alignments, we did secondary structure predictions. In this case, we used this third version of our PCONS method. We made a number of contact maps. We made a lot of structures, and in the structure modeling, in this case, was just using CNS, which is what was called confold. Someone hacked CNS. Basically, the same method as used for using NMR structure prediction. Understand it was just hacked a bit to add in this information. And we generated a pool of models here from this, because there are many different methods, and we had the protein structure at the end. And we applied this to all of PFAM. So PFAM is a family of, it's a database of protein domain families. So at that time, I think it was like 15,000 families in PFAM at those days. It's maybe 20 now. And we took all these structures, all the, I think about 7,500 families that had no structure information. We couldn't model by homology. And we tried to predict it. So at the end, we ended up with 500. So it was not that many, but it was at least 500 new families we could model, give a structure to. At the same time, there was the Baker group did something similar, published more or less at the same time, or slightly, and they had also about the same number. The overlap was not that great because metals were identical, so in total you could maybe do 700 proteins. So we had a bit of, there was an overlap between our methods. So there was, at least there was some novel biology that could be predicted, but still, it was 500 out of 7,500, but still 7,000 missing. And the models were not atomistic quality either. There was a development. I guess the key paper here to describe is the Raptor X. It was the first, or maybe not the first, but the most successful of the more deep learning-based contact predictions. It's based on same ideas we had done in other papers. So you take your sequence and you do these co-evolutionary predictions using DCA or PLM-DCA. Some of these methods exist there. But you also do prediction for each residue. And the trick, so then you get like, if you have a sequence of L, you do N predictions. You can predict the secondary structure, you can predict the surface area, you can predict the, whatever you want from it, basically, and the sequence itself. And if you do an outer product of a vector L, you get an L by L matrix with a depth of N, or 3 times N. So you basically get a square matrix here also. It doesn't really mean anything, but it is something that the machine learning can learn from. And you do that, and you take the co-evolution information that is already L square, with, in this case, they use 3 different numbers. And you merge it together, and then you use basically techniques from image recognition. You use a convolutional network, and you do predictions. In this case, they did it for the whole proteins. You can do it for a small part of the protein. It doesn't really matter so much. But you do 2D convolutions, and basically so the input is something that represents contacts, and the output is a new contact map. And this was in like 2017, 18. It was showing some progress. It was getting, I mean, it was clearly, there was a lot of interest in the field. It was not really replacing other methods, but it was there for the hard targets. We could do predictions that you couldn't do before, and at least with quite high accuracy. So it was really interesting. So I guess that is when DeepMind got interested in the field. And in CASP 13, when in 2018, suddenly DeepMind entered CASP. So CASP is our structured prediction contest every 2 years. And this is sort of like the plot I think I showed a couple of times, where you have a measure here, which is called GTS, which is basically quality. 100 is perfect, 0 is random. About 90 is probably experimental methods accuracy. So it's not nothing. So if you take the 2 structures, the same structure, and do 2 different experiments and compare them, you get 90. And then there is a measure of target difficulty, which is basically how similar is the most similar protein in the database. There's some other measures, but there's a rank that people used for many years. And CASP has been running since 1994. And this is just a straight line, but you could look at it. You see that for the easy targets, the methods has always been good, and at least was a pretty good improvement in beginning, a small improvement, but it's been slightly better, but not a lot. For the hard targets, it took 10 years maybe to jump from something very random to something better random, and then nothing happened for the next 10 years. And then suddenly, between CASP 11 and CASP 13, performance jumped quite significantly. And in particular, CASP 13, it was clear that DeepMind's AlphaFold 1 method worked quite well. So you had some impressive predictions. This you see either the blue or the green is the native model, native, and the green is prediction, and they are quite similar and quite high accuracy than before. If you look at the contact maps, one side is the real contact map, and one is predicted one. And this is not contact, this is distances. So yellow is the shortest, and blue is the furthest, and then dark is more than 20 angstrom away. So they're very symmetrical, so you can see that the predictions were quite good. But still, it was not atomistic resolution. It was not as good as you see. It's still quite far away from the best for the easiest target. And how did AlphaFold 1 do it? And I will actually not tell you. I will tell you how TR. Well, this is the. Yeah, sorry. This is the same. This is just another non-straight line fit. You see the jump here. And this is an example. It's like you see, it's not perfect. This loop here is wrong, for instance. But most of the backbone is quite nice. Side chains are not perfectly packed in this case. So I will not tell you how the AlphaFold 1 works, but I will tell you how TIA Rosetta works, because I have a better slide for that. And it was released by Baking Group 6 months later. It's basically a copy. So the key difference from what we've compared with Raptor X difference is actually that you do a prediction of distance bins. So you do, instead of predicting contact, non-contact, you predict probability of a distance. You have distance bins from 0 to 20 angstrom, and you predict probability of each residue to be in that distance. The input is basically the same. It's the same input. You still use these DCA methods or other methods for predicting things that you then put into a network as an input. And then TIA Rosetta also use some angles. They're not that important, but anyway. But the good thing about this distance is, well, I think there's another thing is that they tell is important, is that this network here is much deeper. So the network used here, first it's used another function a little bit, but particularly it's much deeper. So they do it for small, they will do it for a 64 by 64 block, not the whole protein. And that enables them to do it much deeper. And if you do it much deeper, you can learn much more advanced information somehow. So they really said that the deepness is the important part. And that partly is because GPUs are better, you have more memory, but partly it's also DeepMind's experience in doing machine learning. But the idea is you have these distances, and you can actually turn these distances into potential. You can turn it if you have probability of distance. You can say, oh, this is going to have a peak at 5 angstrom, and it's going to fall off, or you can have 2 peaks, whatever. You can have some distances. And this you can put into a simple minimization protocol. You don't need to have any fancy Monte Carlo or distance geometry. You can do simple minimization in a fold, voice overlap, and get a good model, and then you can add side chains and relax it a little bit. So this method is actually much faster than the early methods. So you do a very quick method there. They tried other tools also, but they really didn't gain anything from it. So it was still, I would say, impressive. They were clearly better than the early methods, as you can see here. There was a jump in performance. And we jumped from there to there. But it was basically just an extension of earlier work. So we sat there and realized, yeah, now suddenly deep learning works. We can use these methods. AlphaFold 1 is not available, but TIA Rosetta was available, and there's some other people that implemented similar things also. So we thought that basically, and we did some work on it, showed that basically it works for almost everything we tried. Like everything in the big family. It's not fantastic accuracy, but it's good enough. So for instance, before we had problems with repeat proteins. You did repeat proteins. And that was a problem with the same methods, because you get patterns that were overlapping and didn't get good models. But these things just worked. You just put it in, you get a nice model. We had some problems where you really had interesting evolutionary information. You had a part of a protein that was missing 3 residues, missing transmembrane helix in the middle, which means that everything else has to turn around. And we could really check, we can run this, we get a perfect model of a new loop forming, and everything turned around, and it looks fantastic, and it agrees with functional ideas of proteins. So you could really use it for some biology. But we said next thing is protein-protein interactions. This is going to be the things. We started looking at that. The problem with protein-protein interactions from this point of view is that you have 2 MSAs, and you need to match them. It's not, you want to look for co-evolutionary signals. So you need to know that exactly this line, this protein in M for protein A, and this line for protein B are interacting. And if you have only 1 copy of each protein in each genome, it will be super easy. They always interact the same way. So if you have always that. But in particular eukaryotes, you have many paralogs, you have many copies. And it can be so that 1 protein interacts with all the other ones, or it's just that you have a particular one interact with another one, and it can change and so on, or in some circumstances interact with the other one, etc. and so on. So that means that it's quite hard to find optimal matching there. And also so what we do, and everybody else does nowadays more or less, is that we just take the first hit of each proteome. We take protein A, we search all the proteomes, we take the top hit in each one, search B, take protein B, search them, and take the top hit in one, and put them together. That means that we throw away a lot of information, so the depth of the MSA is much smaller, but it seems to work. And nowadays, we have a lot of sequences. For most cases, it works. So we did this. very simple principle. We took our MSAs, we merged these alignments, they shrank and become smaller. We used TR Rosetta, because that was available, make the contact predictions. So in this case, you can see one side here is the contact, real contacts, and one side is the predicted contacts. We used basically Rosetta also, PyRosetta, the same protocol as TR Rosetta, but just hacked it a little bit to make 2 chains, and made predictions. And we did scoring. And we found out the best scoring method we had was basically the simple, if you run this many times, if you always get the same answer, you were most likely correct. If you get a different answer, you're most likely wrong. So just looking at consensus between the methods was quite good. So we showed, there were cases where this worked. So this is a perfect prediction that we could do. It's like it's 2.6 angstrom. It was quite nice. But unfortunately, it didn't work in every case. So here you can see we can only get like few contacts here that are actually they might be correct, but most of the contacts that should be there we don't predict. So it worked in some cases, but not at all. At least in Casper 13 we had used this, and we made one prediction that was good that we used for this. That was the best one, but it was for one target, so we were not at all that good overall, but at least it worked in one case. We also found out the same thing again, that like, so this is 2 different proteins here, and they're 2 different alignments. And in this case, this alignment works well. And in this case, this alignment works well, but not the other one. So we didn't know what was the best way to alignment. It was not one way to do it. We had to try different cases. We had to do many alignments. But because the scoring worked, we could do that. But still at the end, we end up with something like, let's see here, this is our quality. If you take the Pico's doc here, it's like the red ones. You see, you have some models that are good, but not so many. And if you compare to other methods, like old classical doc methods, we were more or less equally good or equally bad, I would say. They are like an average score is basically close to 0. There are a few models in these methods that are good. And that's, I mean, this is excluding simple homology modeling. That would be better if you included that, but if you include that. The only thing that was a bit nice is it's sort of a bit orthogonal, so we could at least be a bit, add some information to this. But, I mean, it was a start, but it didn't really work. And then in Casper 14, we saw this. So suddenly, if you look at the hard targets, this DDTTS jumped from like, it was 40 in Casper 12, in Casper 13 it was close to 60, and now it's close to 90. So, and you can also say that the methods that didn't use AlphaFold 2 was basically catching up to AlphaFold 1. It was more or less equally good as AlphaFold 1 was a few years before. So of course we had to figure out how it worked. I mean, yeah, this is also the target, the same one plot again. Casper 13 was the red one here. And see, this is here is Casper 14. It's almost a flat line. So it's almost as good for the hard targets as for the easy targets. And it's almost over 90 all the way. So it almost is as good as experimental methods. So it was extremely impressive. And of course, everybody was eager to find out how it worked. And if you look at the models, I think this is actually probably the most impressive plot. If you look at the RMSD, so the displacement of atoms, for 95% of the residues, you can also have some ends or flop and so on, but take 95% of the residues, all other methods have about 3. This is below 1. And 1 is basically the distance between 2 atoms. So it's quite good. You can see that you have, I mean, sure, this loop here is not absolutely perfect, but like this really, really follows exactly straight on. This zinc binding site is perfect model with the side chains. Even this big protein is not 1 angstrom, but it's much, much bigger protein than anybody could do with this metals before. And it's 2.2 angstrom, James 1.96. See how short, some shifts here, but basically it works. And it even works for some proteins with very few sequences, but they worked hard on that, but it sort of did. So how does AlphaFold work? This took, I mean, luckily, the paper written and the code is available, so people can understand it completely. But of course, there was Minkyung Beck and the people in Baker Lab sort of managed to copy most of it from the presentation. So they could figure it out, but it was not really everything, but most of the things there. But the description is good enough, and particularly the code is there, so everybody can look at it and do it. That's thanks to the open source initiatives. So the first thing you can think about, it threw away everything done before. It really tried to not build on AlphaFold 1 at all. It's a completely new start. Of course, almost every single part of it has been proposed somewhere else before. It's not like not everything is invented from scratch, but there are papers describing different things and doing different things. But and most of it in computer science literature, but not all. But of course, bringing it together is the key thing here. So it starts with an MSA. It has a way of using templates. We can ignore that for the moment because that doesn't really matter, actually, to be honest. It just makes the prediction slightly easier. But it uses an MSA, but it doesn't do any of this DCA or this co-evolution calculation. It uses the MSA directly. And then it also has a pair representation, and then it has what we call the EVO formers. The EVO former is a transformer-based model. I will show you more in the next slide. And it does this 48 times. So basically, that iterates between the MSA and the pair representation. And then it outputs a pair representation, which is basically distances between every pairs, and it has a structure module. To be honest, you could cut away the structure module, and you do almost as good. You could put that into the old method, and it works quite fine, but the structure module is actually faster and slightly better. You can also do a recycling, and it helps for difficult cases. So it's not absolutely necessary. For the easy case, if you have a clear signal, you don't need it. But for the hard case, when the signal is not very clear, it helps. So but that's I will not ignore talking about that. But it's really the key thing is this EVO former. Well, first key thing is this MSA input. So people had done that before. There was at least 2 papers that had done it before, but not very successfully. I mean, they were okay, but not improving performance. Basically, you do the same thing, but instead of having this co-evolution thing, you just do an embedding of the MSA and you predict contact map. So this is actually the wrong slide, but yeah, it's so instead of using this co-evolution, you can put in MSA directly. So the EVO former does that. So it has an MSA, and then it uses the transformers uses a trick that's called attention, which basically tells you this is the important part that you learn what is important. It does it both on the columns that represent the position in the sequence, and on the rows which represent the sequence, which is sort of useful because it actually they actually did with training. They put in some noise, they put in some bad MSAs also sometimes, so it learns to ignore the bad parts. So that was a trick that people realized afterwards when people tried to retrain it. They did not do that, and they then behave differently. And it still works for cases, but it's more sensitive to adding bad things in it. You do this, you basically let the network learn that this is the important positions, and for things that are very far away from each other in structure, not that important because you have no signal, but things that are close are signals, and so on. So you sort of learn co-evolution signals. You take your MSA then, you do an outer product mean, you get a pair representation, and then you do the same thing here, that you sort of, okay, what content is important. And here you use what we call a triangle update. So you use basically every triangle of points are also important. So I and J and K are important. And then you keep on doing that. And you get some kind of pair representation. Of course, this is a hyperdimensional space. You have no idea what it means, but it means something with distances. And you do this 48 times with independent weight for each layer. So you really sort of iterate and learn more and more. You can learn more and more advanced things. And of course, the benchmarks from less than 48 times worked less well. So you get a very good representation of every pairwise distance. I'm going to skip this in the interest of time. The structure module then takes these distances, pairwise, basically treats the protein, I think this is probably, treats the protein as a gas phase. It tells that every residue is a triangle with an orientation, and they have a way to do this without having being equivalent, so it doesn't matter if you have it rotated or so on. And then you basically optimize the correlation or the packet backbone. You don't even need to know that residues are connected, because you know always residue I and I plus 1 are going to be next to each other, so you don't have to tell it that, it learns it. And then afterwards, it also predicts the side chain conformations and packs the protein. And at the end, it has a refinement protocol you can use if you want to. Okay, I should go on a bit, otherwise we will not have time. So AlphaFold was released in summer 2021. It was mid of pandemic. Everybody was starting working on it. So it was like Twitter was full of discussions, and it was a nice way to spend the summer. And this is from a community paper showing just something that was basically done in the first 2 months. I mean, it took a longer time to get it published, but it was first 2 months, and people did different things just running it. You can use a lot of different things. It basically shows you that the models are almost as good as crystal structures. And it can be used for predicting disorder and other things also. This is just a set of papers that a small set of all, but I have used this for other things. We went back to our work on interactions. Basically took the same thing as we did in TRosetta, but just replaced TRosetta with AlphaFold. We took our sequences, we combined MSAs, we ran AlphaFold. And it was also probably Min-Kyung Beck and some others that showed you that you could do this on Twitter. And then like we just told them, because it's a trick in AlphaFold that you actually have a residue number. So if you have residue number 55, and you then just have another residue number 155, then it thinks it's going to be, it's like a chain break. So you just add chains to the residue numbers, and you have the whole, it treats as 2 chains. And remember one thing here, when we do this AlphaFold 2.0, it has never ever seen 2 chains together. It's only trained single chains. We did MSAs. You can actually take MSAs, just merge them together, and you get something like that. Because sometimes you have MSAs that covers both proteins anyway, because they are some fused couples, so you have some bad MSAs. Sometimes you do that. You can do the pairing as we did before. You can do what we call the block. So basically you just put MSA1 and MSA2, and then you put the gaps in between them on the other side. And you can combine these things together. And in this example here, this one did not work very well. This one either. This combined one worked in this case. So we tried all these things, and if you remember, we had some plot before where you had that looked more like this one. You had a few good models, but most were bad. So this is the DocQ score. It's like 0.23 is a good cutoff for a good model. And using Rosetta Fold, nothing that's really worked. Just take AlphaFold 2 straight off the box. We got this plot here, which is like, we suddenly have a few models that are really good. I mean, there are many models that are, maybe 1/4 models or something are better than our established cutoff, a good one. We use the pairing, we do a bit better. If you combine the pairing and the original one, and also with the top ranked one, we did better and better. So then we end up with like 60% of the models that are over our cutoff. And this is compared to anything else before, is way ahead of it. We showed that still coevolution is probably important, because we use the coevolution method to predict here, and we say in the cases where we have no context in the coevolution, and these are DocQ scores, so it's called our predictions, basically all models are bad. Not all, there are a few good ones, but most of them are bad. While if you have context in the middle, they're about half good, bad one. This is another alignment, so you get, so basically there still is a coevolution signal that's probably important for most cases. And these are some examples. I mean, the packing is perfect in these cases. I mean, this side chain is slightly off, but this is probably up to the surface. So it's really completely different quality than what anybody could do before. We also showed you that you can make a score. So we can basically just take, in this case, we took a very, very simple function. We just looked at the interface, counted the number of contacts, and took the predicted quality of this interface. Because one good thing with AlphaFold is that it's not only making a good model, it also estimates how good it is, which is an important factor. It has 2 measurements, one overall and one for each residue. It's called the PLDT value. And that is it's really good at that, and that's important. We have to say, if we put those things together, but they are badly predicted, we ignore it. But if you don't put them together, we don't think it's a good model. So we just fit this simple curve to it. It's perfect, but at least it's quite good. And we showed that this measure could go separate good and bad models. Not so surprising, because that we did. It has an area on the curve of like 0.95. And they can also be used to predicting interacting and non-interacting proteins from our data set. So not as good, because there are some that we probably miss, but at least it's better than other methods to do that. Which means that we could take this, we can skip for the moment. We can take this on large data sets. So we took 2 data sets. One, EuroCC, which is a yeast-2 hybrid data set, which is basically people try to ask to interact, and there's like 65,000 interactions. And one from a co-purification data set from Ed Marcotte and colleagues, which is called UMAP. And then we run this and we estimated this P dot Q values. We remember like somewhere if you're more than 0.3, 0.4, you have a good one. If you're less, you don't know. Good prediction. So if we look at these red curves, that's basically this subset of these data sets where we have some additional evidence. Either you have a structure module, or we have extra cross-linking data, or extra additional yeast to hybrid experiments. And in all of these cases, we have about 60% of the models are good and 40% are bad, which agrees with what we see in PDB. If you take the random data set, which is these blue ones here, we basically have everything has bad scores. So basically, for random proteins, we don't predict any interactions. There are a few one up here. If you take the UMAP data set, it's not as high as the red one, but it clearly is a bump up here. While in the Euro data set, this bump is very, very small. So in the yeast to hybrid data set, we can hardly predict any of the contacts that should be there, but in UMAP, we predict quite many of these. And why is that the case? Well, first we can say we can improve the data set, high quality predictions, more than was done before. So we can basically get 3 times more than you could do by structure. Can skip this. So we can see that one reason we can't do as well in UMAP is because we can only see things that are directly interacting. So if we have a complex of 6 proteins and not directly interacting, we can't detect it. We can only detect it directly interacting pairs. So that's and if you do that in the data set, we get something similar. We get basically that curve set, which is sort of a similar size. Yeah, this is another data set to show the same thing. But we show that this URI data set has a lot more disorder. So it contains many more. So that most likely these are transient interactions. I mean, it can be noise also, but it contains many more transient interactions. And it means that we can't detect transient interactions probably. I can't say if they are just noise, if they are there, but we can't detect them. They're not part of stable complexes in the same way. So that's a drawback of AlphaFold. We can't predict transient interactions. And of course, they have smaller MSAs, but that's because they're disordered. Can skip that. But anyway, we could do that. We could create interaction networks. If you look at this network, you get some interesting points. You can look at that one. This is the proteasome. And here we have a network of basically everything interacting with everything. And you can look at this one here, which is the RAB-GDI interactions. Then only it's a very different picture. It's GDI one interacting with all these RAB proteins. If you look at the structure of this, you have the GDI protein and the RAB protein, and all the RAB proteins look the same and bind in the same place. So that means that this is competitive binding. They can't bind all at the same time because they're overlapping. In contrast, in the proteasome, it's a part of a big complex. So if you analyze these networks, you can learn more things about it. Of course, this was already known in these cases, but we could do it. And the interesting thing we did is like we could actually see that we could build this proteasome by taking subunits by subunits. We took one subunit, which are 2 pairs, and I added the 3rd one, and the 4th one, and the 5th one. The problem was that we had to do it in the right order. It didn't work if you just took every random pair, because they all look the same, and then it got sort of overlapping. Some models were correct, some were pretty wrong, and so on. So if you did the right order, it worked. But then, okay, I guess, we used the Monte Carlo tree search method that basically searches all possible orders and do that in different orders. And basically tries to expand as long as you can for each order. If you do that, you can end up with having a large set, or not all, but like 10-15% of these very, very big protein complexes that we could predict with high accuracy. So in this, and you see they are mainly, I think I have a slide that here. You see they are mainly symmetrical. So if there's some symmetry in them, we can get good answers. For the ones that are symmetrical ones, we only have one that is okay. The rest are quite bad. So for symmetrical rings, you can just start building up here and there. And there's been some other groups doing similar things later with even higher accuracy than we have. Okay, I think that was a good time for you having some questions. So.
Using the symmetrical assemblies. either complementary biochemical information that feeds into the process, or does the approach allow you also to guess in an intelligent way what the possible n equals x could be?
So actually, in the original paper, we assume that we know it. In this paper that's on, we asked our, it's under review right now. We show that this is actually done without it. So basically, what we do there is here. Basically, we just ignore the symmetry. We know what components should be there, and the criteria is very simple. We add components until things start colliding. So that basically, the energy function is super simple. And if it's too much, two products cannot be on top of each other. That's the only thing we check, basically. So, and basically, if it's a closed circle, you can keep on doing it. It works. But of course, in few cases, we get a spiral becoming infinite. So we maybe have some maximum number we stop. But actually, for symmetrical ones, it seems to sort of work. Not in all cases, but this data here is actually without knowing it. So there are cases where we have, sometimes you get one wrong, but yeah. People have tried in other cases. It seems to work in some cases at least, yes.
Yes, go ahead.
Thank you very much for the presentation. Just like opinion or opinion-related question, you mentioned everything about work of interactions using AlphaFold, but now we are moving towards, the field is now moving towards a second generation of deep learning models, in this case protein language models, which now we folding methods like OmegaFold or the one from Meta, now they have unraveled interaction for dark proteins or even protein from metagenomic sequences. Do you think that this AlphaFold will continue? Is there going to be advances with AlphaFold related to this kind of proteins, or do you think that we should move to protein language models?
For interaction, this is actually data. This is the success. This is modeling different omega data skipped into some time. So this have 2, 3, 4, 5, 6 things. This is AlphaFold multimer. So you see you drop a bit in performance. The bigger protein gets, but not too much, man. And the reason is for some reason pentaheteromers are hard for some reason. This is our old fold dock method, so it's slightly worse, but still okay. Here you're down to OmegaFold, ESM fold. So basically it's quite significantly worse. And the reason, if you think, what does a language model do? A language model sort of memorizes the MSA. That's what it does. Sort of, it takes information and knows everything. But it only does it for one single sequence at a time. It doesn't learn interactions. So it can't get attention between these interactions. So it could maybe get it to work if you have, if there are fused proteins somehow there exist that you can learn. So it's not completely horrible, but it's far, far worse. And there are other methods like the topsy-turvy by Cohen and other people that are also significantly worse. At the moment, AlphaFold is clearly the best.
But then do you think that we should change the representation for complexes if we want to apply PLM?
If you have an idea how to do it, yes. I don't know how to do it. Because you can't write in the language models all combination of proteins because it becomes square. So if someone has a good idea to do it, that would be great. I don't know how to do it.
Just in relation to that, so you have to, when you predict the interactions, you know which proteins are interacting or likely to interact, right? So can you actually use sequence information to detect where two proteins would likely interact or not?
Yes, that's basically what you. I think you cannot. This is basically. You're pretty good at saying if they are in a stable, complex interacting. It doesn't work for transient interactions. This is separating interacting, non-interacting protein. And if the data says that you can basically say 60% of the proteins that are interacting, we find before we have any false positives. Basically, that's. Yeah. There are certainly not the transient interactions. There are certainly not parts that are very thick. But if they are stably bound together, we can probably, with 60% accuracy, identify them with very, very few false positives.
And for transient interactions?
I would say it's hard. It's like, I mean, you know, there are a lot of studies showing that you're going to get, if you know the interaction, you model different things, you can probably get a good model of it. But it's, for screening, at least we have not got it to work. You're much, much worse off. I mean, we're trying, but I wouldn't say we are succeeding. It's one of our projects to do, but we're not there.
Sorry, just a follow-up question. Follow-up on my question. Indeed, you mentioned about the protein language model, that AlphaFold is still the best for interaction, but especially because it relies on the multiple sequence alignment. But in the cases that you want to model proteins that have hypervariable sections, such as an antibody, AlphaFold fails in many instances. So here, what would be the solution? Because one of the reasons why protein language appears also for tackling this issue. So
I think yes and no. Yes, AlphaFold, so for the proteins that we cannot model today, one set of them is everything which is, well, you don't have co-evolutionary signals. So that's antibody antigens, and because antigen is fixed, antibody is evolving fast. So you have no co-evolution. The same thing with the host pathogen. If you have a virus particle binds to a human receptor, the virus is evolving very fast, so there's no co-evolution here. These cases are very hard, and they are basically not, I think, no one has been doing very, I mean, some tricks can do it better, but nothing works very well. The results sort of hinted in AlphaFold 3 looks extremely promising, but we don't know what to do. The other thing is, I don't think the language model, I'm not convinced that language model is a trick, because it's very clear if you look at the ESM-Fold paper, is that when the MSA is more shallow, the performance for the language model drops even faster than for AlphaFold. So really, when you're a big MSA, the language model works well, but you're a small one. So the idea of using language models for things that are single sequences is, I'm not convinced it really works. So I think we need something else. We probably need more structure, I would imagine. I don't think the language model, at least in the current state, is not the way to get there.
Okay, I have a question too. When you apply, these methods, especially AlphaFold, to predict proteins for which you have no structural information, and you would like to have proteins which we have in the AlphaFold EB. So what kind of confidence metrics, and how can you use the confidence metrics to really prioritize these models, and how reliable is this?
I think they are. For individual proteins, the scores are really good from AlphaFold. Which is, I think, like machine learning methods is fantastic. It really methods that tell you when it does wrong, when it does right. So, like for single proteins, you can look at the average PTTM or the PTM or average PL. That is, of course, and it's also known that when it's bad, it's most likely because it's a disordered region. So that's for the complexes they have measures for that. They have what they call ranking confidence with the IPTM score. It's only for the whole complex, so it doesn't tell for each side chain. We have a score for each subunit. That is maybe slightly better, but it's overall it's okay. But one of the tricks that people do is that they generate many models. You run 5,000 AlphaFold models. And in our case, it has some correlation with the quality measures, but there are cases where it says that all models are equally good, but there are a few ones that are right and all the others are wrong. So it's not the perfect measure. That's another thing we are trying to do better, but we are not succeeding yet, is to improve that. But so it's not, for that it's for the
physics-based methods to improve this?
Maybe, you could use some post screening is possible. That's one way to do it, yeah. We haven't tried that exactly, but yeah, that's one way to do it. That's the question over there.
Thank you for your nice talk about protein-protein interaction in AlphaFolds. I was wondering what's the status of AlphaFolds, because sometimes, like in the case of kinases and also GPCRs, you have active and inactive states of the proteins. Do you think that AlphaFold will be able to also model those different states of the proteins?
So there is quite a lot of work on doing multiple states. In many cases, if it's in the database, it can do both things. And if you animate model models, you can actually also go both states. But of course, it's not I don't think anybody knows which one becomes more frequent or not. But it has the trick is to generate many models, but you can do it. Basically, you can just run it many times. You can do dropouts, and you can play around with the MSAs and things like that. It's a bit unclear what is the best method to do it, but it probably doesn't really matter. But you generate many models, and often you can get different states. It's not really clear if you have things that only have one state, if you also get different states, and how to separate. I think people have never really done the negative tests. But there are, for active and inactive forms, if they are in the database or if there's nothing very strange about it, you often get both of them. So, and sometimes you get a third state that you think could make sense, but it's not in the database. And, well, that's it might be right, might not be. So you have to use your biological knowledge there, I think. Yeah.
You rightly say that it's an important area of
Yeah.
active research to try to sample enough and to try to generate multiple confirmations. And it's going to be an area where most developments are going to happen in the future.
Awesome.
Yeah, I'll move closer to the microphones here. We have a couple of questions from people joining online. The first one is from Giovanni Mazzocco, who's asking what would be the optimal scores to evaluate the likelihood of interactions like PLDDT, PDOQ, etc., or combinations of them?
It's slightly, the thing is that it slightly varies from version of AlphaFold. If you remember, the PDOQ was the AlphaFold 2.0. And that's, you didn't have any other scores. So it works quite well for that. And it's still some indication that that is the best way to measure interaction on interaction. That the multimeter version of AlphaFold sort of brings things more together. So that's, we haven't really gone a careful benchmark, but Minkong Beck has done that. And it claims to be the best way actually to use the original monomer version to separate interacting and non-interacting ones. And otherwise you can use, you can use the built-in scores are fine, but you can use some other ones. There's not a clear, very big difference.
Okay, then I'll move on to a question from Melvin Young, who's asking, does AlphaFold already include all the known ways to predict contact maps, or is there more to be added?
Mm-hmm. I'm sure there are new machine learning tricks that can be used. But there's no ways to do it. It's really machine learning, and it's just one machine learning model used. The Rosetta Fold is slightly different, and the latest version more or less as good. And there are Omega Fold has their own slightly different versions, but they are all inspired by each other. But I'm sure someone will come up with an even better idea in the future.
Okay, then I'll take a last one before we go into the break. So the last one is, can we leverage biological assemblies as template for accurate multimodal predictions in AlphaFold? Yes, you can use, include templates. Default AlphaFold does actually only use template for monomers. So each template, but you could actually hack it and use it for multimeter there also. So there are, I think people are not using it really for so much for structure, but if you have like a low resolution cryo-EM data, you don't want to put it there, or you want to bias a sampling, you can do it. But it demands a little bit of a hack. There are hacks available to do that.
Okay, thank you very much for a nice talk and for all the questions and also for the online questions. And we have a present for you from the organizers, which I'm sure you would enjoy. Something representing Belgium.
I will enjoy, yes.
And we have a break now.
A break.
Short break, be back in a little over 10 minutes.
Okay, perfect.
So insane, I believe that you walked away. Oh my, oh my, oh my, oh my. Golden days are coming back again. When you showed up, I felt like a different man. Remember yesterday, oh my, oh my, oh my. for you right away. Oh my, oh my, oh my, oh my. Show you around the town, and we watched as the rain came down. The rain came down. Golden days are coming back again. When you showed up, I felt like a different man. Remember yesterday, oh my, oh my, oh my. Whoa, oh, 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 oh. Whoa, oh, 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 oh. Deja, deja vu. Oh, 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 oh. Whoa, oh, oh, oh. Deja vu. Deja vu with you.
I've got to get some sense back into my head. I'm in the dark, and I can see where I'm being led. And I give the world to set the clock back and act like a man. Where can I turn to save myself from this confusion? I walk in the park.
drive home to you, baby. I'm faded. All I wanna do is take you downtown, baby. I'm wasted. Baby, I'm faded. Baby, I'm wasted. All I wanna do is drive home to you, baby. I'm faded. All I wanna do is take you downtown, baby.
infatuated, feel the power in your eyes. Must be voodoo you're doing, it's pulling me back. Does it sip of your potion in my body?
Okay, this second talk is by Casper Goverde, and he is at the EPFL now in Switzerland. He's a young researcher in the lab of protein design and immunoengineering, and this lab is headed by Bruno Correra, and they are doing really nice work at the forefront of applying deep learning methods to protein design. So please, the floor is yours.
Thank you very much for your introduction. I'm Casper Goverde, and I came here yesterday by train from the beautiful city of Lausanne. So this is an overview of our campus, and we are located in this building right here. And I am a member or a PhD student in the Laboratory for Protein Design and Immunoengineering, LPDI, under the supervision of Professor Bruno Correa. And today I'm going to be talking to you about the design of soluble analog of integral membrane proteins, because this is what we do. We are protein designers. So let's first have a quick overview of what membrane proteins exactly are. Generally, when I talk about membrane proteins, more specifically, I talk about transmembrane proteins, which come in three different types. You have the single alpha helix, you have the multiple alpha helices, and then you have the multiple beta sheets. So generally, these proteins, they insert into the membrane, and in the case of multiple beta sheets, they form a barrel-like shape to form pores in the membrane, which exerts exact control over the fusion of small molecules over the membrane. And generally, these multiple alpha helices, what they do, they perform functions as receptors. So they can interact with other proteins or biomolecules, change conformations, and activate some kind of signaling pathway. Even though only 1/3 of the human proteome are membrane proteins, more than half of the drugs target membrane proteins. So they're very important. And besides that, there's also something funny going on, where with regular proteins, when it's being produced by the ribosome, you have your hydrophobic and your hydrophilic residues. What generally tends to happen is the hydrophobic residues, they tend to cluster together, and this is the main, the driving force behind protein folding is the hydrophobic effect. So you can think of this as having a drop of oil in water. This drop of oil tends to stick together. It doesn't like to interact with water through the hydrophobic effect. So a soluble protein, what we generally see is we see a hydrophobic core and a hydrophilic surface. So it becomes soluble and doesn't aggregate. However, with membrane proteins, we kind of see the opposite, because it's inserted into a hydrophobic membrane. So what we observe here is that we have hydrophobic residues on the surface, which generally interact with the membrane. And in the core of the protein, we have this weird mix of hydrophobic, hydrophilic residues, because all of a sudden it can create this complex hydrogen bonding network in the core, because there's no water around, so it can't be disrupted by water. So now all of a sudden this is the main driving force, or one of the driving forces behind protein folding. And besides that, when we take a look at the SCOP database and we take all the different protein families and we take a look at the soluble proteome versus the insoluble proteome, we find that some protein folds, like the IG-like fold and the TIM barrel, they are unique to the soluble proteome. You will not find them in the membrane. Some proteins, like the beta barrel, you can find them in both the soluble proteome as in the insoluble proteome. So you do find some beta barrels in the membrane. And then there are these unique unique topologies, such as the GPCR, which are exclusively found in the membrane. And this raises some very interesting fundamental questions. Why don't we see these topologies in the soluble proteome? Is it because the dynamics in the membrane are so different that these proteins they cannot exist in the soluble proteome, or is it just that these topologies they have not been explored by nature, or there has not been an evolutionary pressure or the need to develop these protein topologies in the soluble proteome? So yeah, we are protein designers, so we thought, okay, can we design some of these complex topologies of both the soluble proteome and the insoluble proteome and design them as soluble proteins? And especially when we focus on the insoluble proteins, we can answer the question, okay, maybe we can get these topologies from the insoluble protein to the soluble proteome and have all kinds of different useful applications for them. And since this is a talk about AlphaFold, we're going to talk about AlphaFold. So 2 years ago, I was 1 year into my PhD, and then AlphaFold dropped. And the first question that we asked ourselves is how can we use AlphaFold for protein design? So one thing you can do is you can invert the AlphaFold network to do protein design for you. And one way to do that is through error backpropagation, which I'm going to explain now. So generally what you do is you take a random sequence, which is a one-hot encoded matrix. So this is length 20 by the length of the protein, because you have 20 different amino acids times the length of the protein. And this is what you input into AlphaFold. And this perfectly fits with the talk Arne just gave. So this goes into the EVO former, then goes into the structural module, and this predicts a structure. So if we start with a random sequence, we get a random structure. Now what we can do is we can take the structural loss and calculate how far off we are from the structure we want to design for. So in this case, it's the phase loss that AlphaFold uses. So you can think of this as an rotationally invariant RMSD. A more accurate version of it. And then we calculate the derivative of the input over the phase loss, and this gives us an error gradient. And again, this is 20 by the length of the protein, so it tells us which amino acids to change and what to change them into to come closer to our target structure. And we track this error gradient in a PSSM, which is a position-specific scoring matrix, which is again 20 by the length of the protein. And you can think of this as a probability distribution that we're keeping track of. That is, again, for each amino acid or for each position, we keep track of the most likely amino acid that we can find there. So we sample from this PSSM and we iterate this. Basically, this is exactly the same way how you would train a neural network. You would have a loss function, you would calculate your derivative of your input, but then over your weights, and you would update your weights. However, what we do is, so when you train a neural network, you fix your input and you allow your weights to be updated. We do the opposite. We fix our weights and we allow our input to be updated. And in machine learning, this is generally called network dreaming. I don't know if you've seen the images, but if you ask a neural network, like what does a basketball look or what does a human face look like, and you see all these horror images of what a neural network thinks it looks like, it's exactly the same thing. In protein design, we call this hallucination. So we let AlphaFold hallucinate proteins. So first thing we did is, this is also not a new idea. This has been done before, especially with TR Rosetta, so the same method that Arne mentioned in his previous talk, but it hadn't been applied to AlphaFold 2 yet, so we tried to be the first. So we took several different protein topologies, the top 7, the protein A, protein G, ubiquitin, and 4H. And we put them in AlphaFold and we designed sequences for them. And they more or less all looked like the protein I'm showing right here, where we colored in red the hydrophobic residues. So if you remember from the protein folding 101 slide I just showed, generally you would expect hydrophobics in the core, which there are, but here there's a lot of hydrophobics on the surface. So this doesn't make sense at all. So these proteins designed by AlphaFold, none of them worked. Even though they had great PLDDT scores, and AlphaFold would think that they were folded. We have several reasons to believe why AlphaFold is such a horrible designer for proteins. One of them is that during training, 10% of the input sequence is masked out by AlphaFold, which makes AlphaFold very robust, but also very a horrible sequence designer, because you can get 10% of your sequence wrong and AlphaFold will still fold your protein, in theory. And the other thing is that AlphaFold has no idea about protein surfaces. Like Arne mentioned in his talk, you only show single chains to AlphaFold. So AlphaFold doesn't know what a protein contact is, a protein-protein surface, what it looks like, or what a regular protein surface looks like. So these are all the things that could go wrong. So we decided to use Rosetta, which is a protein design tool, to redesign the surface to become completely hydrophilic. And then some of the designs started working, but still it wasn't great. And in the meantime, a new tool came out, which is called Protein MPNN. So now our pipeline is updated a little bit. So we still have our AlphaFold backpropagation, which generate a bunch of different variations of structures with sequences. And then we have Protein MPNN. And what Protein MPNN does, it takes an input, it takes the backbone as an input, converts it to a graph, and it takes a look at the nitrogen, the C alpha, and the carbon atoms, and then it performs something called iterative decoding. So it first encodes the structure into the network, and then step by step, a random residue is picked, and the network is asked, what is the most likely residue you think should go here? And then it does this again and again, and each iteration it starts assigning residue identities. And this actually worked really well. This was shown by the Baker lab that all these AlphaFold generated designs, they can be rescued through Protein MPNN. So Protein MPNN generates a pool of designs. So the way how I see it is we use AlphaFold to generate a bunch of different backbone structures, then MPNN to generate a bunch of different designs, and then we put them through AlphaFold and we select our best designs based on RMSD, so structural likeliness, structural confidence, so the PLDDT in this case, and sequence diversity. We don't want to sample structures that are only 1 point mutation away. We want to really sample a diverse part of the sequence space. But now you can ask me, but Casper, why do you even use AlphaFold? Why don't you use Protein MPNN from the beginning? That's where I need this slide to explain this. So here on the y-axis, you see your sequence recovery. And on the x-axis, you see different methods. So if we take our crystal structure, we get about 80% sequence recovery in the core, but only 30% sequence recovery on the surface, which kind of makes sense, right? Because the core of the protein is tightly packed, and there's only a certain amount of puzzle pieces that you can put there to finish this puzzle. Which is actually something we definitely do not want with membrane proteins, like I just mentioned, because if it's going to recover these hydrophilic residues in the core, then our protein is not going to fold. Then we use a backrub and an MD simulation. So backrub, it kind of wiggles the backbone atoms a little bit. An MD simulation, it does the same, but more physically realistic, I would say. But we still get very high sequence recovery in the core. But then when we use AlphaFold, it really starts redesigning this core of the protein. So when we just use the AlphaFold backpropagation, we term AF2seq. We get a completely different core, and when we then pass it through MPNN, again, we're going to be about 20% sequence identity. So it's a completely different protein. All right, so now we're ready to redesign some membrane proteins. So in this talk today, because of time constraints, I would only like to focus on the claudine, which is a protein that interacts with other cloudines through these beta sheets that stick out, and they form tight junctions. So this is how cells stick together and form a semi-permeable layer of cells. And I would like to focus on the GPCRs, which is one of the most important receptors in the human body. So initially, we were just playing around with taking these membrane proteins and redesigning them, and then something kind of expected but also unexpected happened, is when we put it through AlphaFold and we redesigned it, it started redesigning it as a soluble topology, which kind of makes sense because AlphaFold is trained on the protein data bank, and the protein data bank, even though a third of the human proteome is membrane proteins, only 2% of the proteins in the PDB are membrane proteins because they're super hard to crystallize or determine their structure. It's become easier now with the arrival of cryo-EM, so I think it's a bit more than 2%, but I need to check. But then when we take this structure of the GPCR and we put it through protein MPNN, it all of a sudden it's redesigning it again as a membrane protein. And here, the red dotted line, it is the wild type GPCR that we picked. So it's really making it hydrophobic again, which makes sense because protein MPNN, it was trained on sequence recovery. So it's completely overfitted this model. All it wants to do is recognize what fold it is and just remember what it has seen before. So one trick we did is we took MPNN and we added a bias on the decoding step. So we added a bias towards sampling hydrophilic residues. So you see that there's indeed a shift of the distribution towards a more hydrophilic protein. But still, we weren't impressed. And now it happened that Martin and me, we were at RosettaCon. So Martin Pachessa is the other person on this project, and we spearhead this project together. And we met Justus de Paris, who is the first author of the MPNN paper. And we were having a beer at RosettaCon, and we discussed the idea like, hey, can you maybe retrain this model for us, but without any membrane proteins in the training set? So yeah, I think this is one of the great success stories of why we should still have in-person conferences, and also why we should have free beer at conferences, because good things can happen. So yeah, we got soluble protein MPNN, here termed protein MPNN sol, and what you can see is indeed now we have very much a distribution that is generating very soluble proteins. So we selected our best designs based on confidence, RMSD, and sequence diversity, and we ordered them. So as you can see, only one of the AlphaFold sequences kind of worked. So it was expressing in soluble, but unfortunately, I don't think it was folded. MPNN bias actually did surprisingly well for the rhomboid protease fold and for the GPCR-like fold. But for the Claudine fold, here termed CLF, it wasn't great. But the soluble protein MPNN, it got on average a 70% success rate. So this did really well on all of the protein topologies that we tested. So yeah, we tried it. We took some of these most promising designs and we seeded for crystals. And indeed, we got crystals within 3 weeks. I don't know if other people have noticed this, but we have noticed that if we make designs with protein MPNN, they crystallize like crazy. So either I'm the luckiest PhD student around, or there is really something about this protein MPNN that makes really easily crystallizable proteins, which kind of makes sense, right? Because it's trained on protein crystals. So it might unconsciously or unexpectedly design for good crystal contacts. So here we see the crystal colored versus the design, CLF4 in gray. And we got sub-angstrom accuracy of the C-alpha atoms. So we were really, really spot on, which was super surprising for us. And this becomes also very obvious when we take a look at the full atom RMSD. So as we also take the side chains into account, we only get 1.3 angstrom RMSD, which is crazy. And this can be observed when you look at the inside of the protein. You see it's really, really just spot on the prediction and the crystal. And on the surface, there's more variation, because on the surface, of course, you have more degrees of freedom. So this is definitely harder to predict. So this makes sense. And when we compare it to our initial fold, we're about 3.6 angstrom away. So here in colored we have our crystal, and in gray we have our target fold, which is the crystal we used as a template to design this, this structure. And we got the same with the rhomboid protease, but also with the GPCR. So we got a GPCR-like fold, which had sub-angstrom accuracy on the C-alpha atoms and 1.4 angstrom accuracy also with the side chain. So again, what we observed in the core, it was spot on. On the surface, a little bit less so, but still very, very close. And we were about 3.5 angstroms away from the GPCR we used as a target template. So this was what the original membrane protein looked like we used as a template. So if we look at the surface, you see here in brown the hydrophobic surface, and in blue the hydrophilic surface. This is what it looked like before, and this is what it looked like afterwards. So we really designed this transmembrane part, which is usually in the membrane. It's completely soluble now. Big disclaimer, this is not a GPCR that we designed. This is completely non-functional. This has more in common with a brick than with a GPCR, because you can boil this protein and it will not unfold. It is, just to make that clear. But now this is, of course, the next step is, can we bring back function to these things, right? Because it would be great if this is a tool for solubilization of membrane proteins. So let's start with baby steps. So the first thing we did is we took the granulin receptor, which has this loop to which we know an antibody binds. So we actually used fold-seq to find something similar to our GPCR-like protein crystal, and we got a hit with the granulin receptor, and we took this epitope from the granulin receptor from which we knew the antibody binds. We put it back on the or we literally graft this, which basically means we transplant the sequence back into the GPCR, and we predict with AlphaFold back whether this protein still looks okay. And we pick our best designs, and we're going to test them in vitro, whether this now starts binding the antibody. And this is indeed what we saw. So we tested several different designs, and all of them, they start binding the antibody when we transplant back the loop. So this is the SPR experiment that we did. So here on the y-axis you see the response, and generally when something starts binding, you get a peak in the curve here. And the steeper the curve, the higher the K on, and the slower it detaches here, this slope here. The less steep it is, the higher the K off. And yeah, we got about around micromolar affinity for our transplanted back designs. So this could potentially be a tool. So you can think that if you have this GPCR that is soluble and looks very much like a target GPCR, that you transplant back a loop, and you can use this for example, antibody development, which becomes a lot easier than when you have a membrane protein stuck in the membrane, because it's super hard to work with. First of all, you get low expression levels because there's only 2D expression in the membrane, right? Whilst solubly there's 3D expression. Secondly, because of the hydrophobic nature of membrane proteins, they're just really hard to work with. So now let's go a step further. So these GPCRs, they don't just have loops that antibodies can bind to, but they're actually a lot more complex. So G proteins, they are these 7 helical bundles that are stuck in the membrane, and then they have G protein which attached to it, or G protein nearby, which exists out of G alpha, G gamma, and G beta. Now, generally, what happens is you have a small molecule that activates the receptor. GDP gets replaced by GTP. And then a signaling pathway is started, because the GPCR, it has a small conformational change that allows G alpha to start its signaling pathway and G gamma and G beta to start its signaling pathway. I don't know if there are any GPCR experts in the audience or any caffeine experts, because this is a caffeine molecule. In reality, caffeine does not activate the GPCR receptor, but it actually inhibits the GPCR. I only discovered this after I made these slides. So this slide is wrong in terms of the small molecule that I'm using, but the idea is still there. So in reality, it more looks like this. So we have a GPCR in an active conformation and a GPCR in an inactive conformation, where in the active conformation it can bind G protein, in the inactive conformation it cannot bind G protein. And generally, their RMSD is between 3 to 5 angstrom between the active and the inactive conformation. So you see here that this helix can kind of open up to allow the G protein to bind here. In this case, this is not G protein, but this is something called G mini. So it's a synthetic construct of G protein that is just way easier to deal with and way easier to do your experiments with. So this is what we are using. So you have your G protein in its, you can think of this as an equilibrium reaction. You have part of the G protein in the inactive conformation and part of it in the active conformation that can bind G protein. And without the presence of an activating ligand, the equilibrium is going to lie all the way to the left here. So the GPCR is going to be inactive, but in the presence of an activating ligand, it's going to move the equilibrium all the way to the active state. So now baby steps. Can we design, do we have the accuracy to design with AlphaFold the active and inactive state? So can we design a GPCR that can bind G protein designed in its active conformation? And can we design the inactive conformation that does not bind G protein? So we used the following pipeline. We selected a CXC2 receptor with the main reason is that we have a crystal in the active and inactive conformation, so we know exactly what it looks like. And we fix the motif, so we fix the residues, the amino acids that interact with the G protein. Then we put it through the AlphaFold 2 MPNN pipeline whilst keeping these residues fixed. And hopefully we should end up with a soluble GPCR. It's not a GPCR because it cannot bind any ligand. All it does is it binds G protein. So we will test this in vitro. Now, secondly, can we design for the active conformation, also keeping the same residues fixed, we keep fixed for the inactive conformation. And we put it through AlphaFold MPNN, and then we should get designs that start binding G protein. So we will test this. So what we would expect is that we get an active conformation that can bind G mini and an inactive conformation that actually does not. So we actually did these experiments last week, so I'm very excited to share with you the results. And this is indeed what appears to be happening. So we have our inactive conformation of the GPCR-like fold 13, where we fix the G protein binding residues. And here we see when we do an SPR experiment, indeed no binding curve. But when we design the active conformation and we test for G mini binding, we get a response. So this is very exciting. Even though the binding isn't great, it's about 36 micromolar affinity. It's unmistakably that there is binding. However, it's hard to compare to the wild type because we don't really know what the wild type affinity is because generally G proteins, they're anchored into the membrane. So it's hard to compare. So now we know that AlphaFold is capable of designing the active and inactive conformation. So next thing that I want to be working on is indeed, can we make this protein now switch states? Can we find a sequence that can occupy both states? But that's next. Now finally, I would like to end with the claudine protein. So claudines, they're inserted into the cell membrane and they form these huge complexes. They can interact with other claudines and they form tight junctions, which are semi-permeable, I would call them semi-permeable complexes that only allow a certain type of biomolecule to pass. So this is what they look like. They're 4 helical bundle with 5 beta sheets that interact with one another. And these claudines, they can interact with something called enterotoxin. And enterotoxin is a protein that is being produced by a lot of bacteria like Streptococcus. So you have this bacteria, for example, in your gut, and what Streptococcus likes to do is produce this enterotoxin, and then your tight junctions, they open up, and it can slip through, and if it's lucky, it can steal some of your the resources your body worked so hard to generate. Then what it wants to do is produce more enterotoxin and open up your blood vessels, go into the bloodstream, and produce even more enterotoxin, open up the blood-brain barrier, and enter your brain. So it's generally not a protein you want to have around. But we tried to solubilize these claudines, and we wanted to test if this could maybe be a screening method to check for enterotoxin binding, to see which types of claudines bind enterotoxins. So we took our human claudine 1. We actually tested it on human claudine 1 and human claudine 5, if I remember correctly. We fixed the functional motif again, so these are the residues that interact with enterotoxin, and we redesign the 4 helical bundle part with AlphaFold and MPNN, and we select our best designs again. And finally, we do our SPR experiment, and we do indeed find that that these designs now, they start binding enterotoxin. But not only do they bind enterotoxin when we fit, I know this is a horrible fit, but when we fit our KDs to this, we find that the affinity actually is similar to wild-type affinity claudines. So this was pretty exciting to us. Yeah, these fits look horrible. So one thing we noticed what protein MPNN is doing is that it's making very charged surfaces. So somehow we think that the claudines are sticking together for some reason, and that there's something else going on. So we're trying to figure this out. Yeah, we just recently got this data, so this is not ready for publishing at all, but it's unmistakably binding with similar affinity to the wild-type protein. So to conclude, what we have now is a generalizable approach to various membrane protein classes. So this should be applicable to many different protein classes to solubilize them quickly and easily. And more importantly, we can just do this in E. coli. We don't need any eukaryotic cells to work with. So all these proteins, they were expressed in E. coli without any special buffer requirements or refolding of the proteins. With very high accuracy and success rates, E. coli is generally easy to work with. So what can we do next? So the next thing that we would like to do is try to introduce conformational switching. So we have shown that AlphaFold can design for the active conformation in GPCRs and the inactive conformation, but can we now maybe design a sequence that can occupy both states? Which would be really cool to start designing a synthetic GPCR. Next thing is instead of fixing the G protein binding site, we can fix the ligand binding site. And this would be very interesting for the pharmaceutical industry, for example, because what they do is they take these receptors from the membrane and then they screen it against a library of 1,000 different components, and they see what kind of chemicals would bind to this receptor, and this could potentially be used as a new drug. So what would be easier is if you solubilize the receptor, keep the ligand binding site fixed, and then use this for drug discovery. It's easy to get a lot of protein in high concentrations, and generally soluble proteins are easier to work with. We're actually testing this part of the pipeline next week. We made a bunch of designs for ligand binders, so GPCRs that bind both peptides and small molecules. So I'm very nervous about next week, so fingers crossed. I hope that that goes well. Then thirdly, now it's time for me to hallucinate a little bit. We can go even further. So one of the very interesting GPCRs is a GPCR called rhodopsin, which can be found in ice. So you have rhodopsin that has a small molecule in there, which is sensitive to light. So When it gets hit with light of a certain wavelength, it changes conformation, which allows the GPCR or the rhodopsin to change conformation, which starts activating a signaling pathway. So can we solubilize these rhodopsins and then have some kind of optic switching effect, which would be really nice and very useful for many applications. And finally, vaccines and mimetics for antibody generation, so we can transplant back extracellular domains of these receptors and then use these for antibody generation. For example, you can use them as vaccines, so antibodies are being created against certain epitopes of this protein, or for example, you can immunize mice with them, and then take the antibodies of mice and have antibodies that target a specific GPCR in the human body as a therapy. So yeah, first of all, I would like to thank my professor, Bruno Correa, who's helped me get all this research on track, and most of all, Martin Pachessa and Nicolas Goldbach, who are with me on this project and are carrying their weight just as much as I do. And yeah, I couldn't have done it without these people. And as a special thanks to Alex Vecchio, who is an expert on the field of claudines, who helped us with the binding assays of claudines to the enterotoxin. So yeah, thank you very much for having me.
Thank you for sharing with us these exciting results and this headway into the unknown. Do you have questions? I'm sure. Yes, please.
So my question is, I like the presentation. I just wondered, so many of the things you do is still based on the extracellular part and not really on the membrane part.
Mm-hmm.
And with diffusion-based approaches, you can also do the same thing, right? So that you only focus on the extracellular part and the rest you in paint or scaffold with the diffusion there.
Yeah.
So I wondered, where do you see the advantages of your approach versus a diffusion approach?
Versus a, sorry?
A diffusion-based approach.
Fusion-based approach. Yeah, so I would say that these are really baby steps. First, we're going in as aggressive as we can. We redesign as much as possible because we know this works. But eventually, we would like to see how far we can go back to the wild-type protein again to keep as much residues conserved as possible. So hopefully, it will start behaving more like a wild-type membrane protein, if that's even possible, because you're taking it from the membrane, right, and you're putting it in solution, so your dynamics are going to be completely different. So I don't know. This is one of the questions I'm very excited about.
So what you say is actually that it will be more native, because in diffusion you can to more or less adopt the shape that you say the amount of amino acids, the amount of genes. Can I ask you a second question, or should I?
Go ahead. Exceptionally, yes.
Just short. Another thing is, so we are working a bit on the same with the diffusion-based approach, and one of the things is the impact of lipids. Because lipids are not just around your protein; they also have an active function in the protein. And I wondered, how do you deal with that?
Yes, exactly. So to be clear, we are not solubilizing membrane proteins yet. We are really trying to design it bottom up. So as I understand, you're trying to come from a top-down approach. We are trying to come from a bottom-up approach to really understand through design. But yeah, I completely agree with you. When you take away the membrane, your dynamics completely change. So I don't know how fair it is to study function like this with a redesigned membrane protein.
Important limitations and a challenge.
Exactly.
Of how to the dynamics should be similar. I mean, at least allow the same type of.
Exactly.
I just want to say, really nice work.
Yeah, it's very.
No, I appreciate the questions. Yeah.
Yeah.
Can you compare the sequence of the active and inactive GPCRs that you designed that can bind a G protein?
Yes, they look nothing like the wild type.
Okay. And how sure are you that the inactive, the one that cannot bind the G protein, how sure are you that it's not just a very bad AlphaFold model?
So we're trying to solve the structure. That's the only way to know for sure. For the active conformation, we're going to introduce some point mutants that should be deleterious. So we make some point mutants that should break the interaction of G protein binding to make really sure that it's binding in the spot we want it to bind. Yeah.
Can you try to input in activate? You know, you have these predictors that can tell you how activated your GPCR is, so it gives you like a percent of the activation rate of your GPCR?
No, I'm not aware of that. We should talk after.
Yeah. If you had these applications in mind, but I don't understand from the logical point of view, why did you allow these functional sites of the protein to just hallucinate to something else if you knew that you will bring these back to functional at the later step?
To bring back complete function. Yeah, so it's really just baby steps. So one thing we noticed is if we keep too much of the natural sequence there, AlphaFold cannot predict the structure anymore without an MSA. When we design it with AlphaFold, it can predict the structure. So it's also a limitation that we had at the time. Now we have some tricks to avoid this from happening. So we can more and more walk back to the wild type sequence. But it's also kind of, we didn't have a choice from the beginning. And originally, when we started this project, the question was, can we design these topologies? There was not even a question of function. Yeah. It was really from a protein design perspective, which is, I guess, different from a biologist perspective.
So one last question here.
Thank you very much for your presentation, and also I want to congratulate you because it's a really promising tool. I was wondering whether is it possible to combine genetic information to have a better overview of the transmembrane proteins. As you mentioned in the first slide, transmembrane proteins are really relevant in medicine and also in pharma because we have the pharmacogenomics. Most of the metabolizing proteins are transmembrane proteins, but also the cardiovascular diseases. There's a lot of mutations are related to coronary artery disease that are related with LDL and APOB receptors. How can we use this tool for designing better proteins or better drugs for diseases or for screening individuals, either if they are slow metabolizers or rapid metabolizers, or even characterizing loss of function variants?
That's a very, very good question, which I'm not sure if I have the answer ready for you there. But one of the things is if you can solubilize these proteins, and I'm not sure if we can with LPL proteins, if they are solubilizable, but if it is possible, they generally behave easier and they're easier to do drug screening or to find binders for these proteins, or even to study structure. Some of these receptor or some of these very hydrophobic proteins, they're very hard to crystallize, and just by mutating a few residues, it might become easier to crystallize and search for function. But yeah, I don't know. I don't know yet. I haven't given it thought. Good question.
I'll take a last question from the chat. So, question from Melvin Young, who says, "Thank you for the great talk. What do you think is needed to hallucinate with less guidance, meaning without copying a sequence, but just by specifying what you want it to do?"
Without less guidance? I guess, let me just answer the question by showing this slide. So I call this the current state of the art pipeline, which it probably was 6 months ago. And now it's completely different, right? So probably this first step, this AlphaFold 2-step, I should replace with something like a diffusion-based model, where you fix the epitope and then diffuse a structure around it. And I think that's probably going to be the next iteration of the pipeline. That's how I would go for it, and then it becomes a lot easier to design with higher accuracy.
Okay, thank you very much again for the question and for the wonderful talk.
Thank you.
And we also have a present for you.
Very nice.
Thank
you.
better than
Very
the
nice.
Swiss chocolate and the
I don't know. I don't know. Do you need an adapter or?
I'll give an adapter.
Yeah.
All right. Thank you.
You're putting your laptop?
Yes.
Okay.
Good. Yeah, I guess we just. Okay. Yes. 1 second. Okay.
Okay, while you're setting up your laptop, this is Maxim Tsenkov, and he's a young researcher working on the development of the AlphaFold DB, the EBI, EMBL.
It's okay if you don't.
And you know, all of you know about AlphaFold DB.
Ah.
And it's the database that has now over 200 million predicted structures. And it's very important to try to annotate them and evaluate them and try to extract functional information, etc., etc. So he's going to talk to us about this work, which is done in collaboration between the EPFL and DeepMind.
Okay. Yeah. How do I connect the? I was connected to the Wi-Fi. No. Is it just the HDMI, yeah?
Someone help him or something?
I don't know if I need to.
Look.
Or do I need this?
It's up there.
Sorry? Oh, there it is. Oh, okay. Cool. Okay. Gonna have to do this without notes. Okay. Sorry about that. I have given slideshows before, I promise. Okay, cool. Yeah, so thank you everyone for coming, and thank you for the invitation and for organizing. Yeah, my name is Maxim, so I'm one of the bioinformaticians at AlphaFold DB in the PDBE team. And yeah, so I just want to, I think everyone's pretty familiar with AlphaFold by now, and I think I wanted to kind of focus on what can we do now that we have all this data. And I'm going to show you some of the features that we're working on as part of the database, and hopefully support some of the use cases that you may or may not be doing. Okay. I'm going to cover for the first bit about AlphaFold, but Arne conveniently covered some of it, so maybe save me a few minutes here and there. But really, I want to kind of dive into a lot about how to, as I like to phrase, navigate the structural landscape. And some of the things we're doing, so we're integrating this tool called FoldSeek, which I'll tell you more in a bit, and kind of show you what you can do now with integrating these, this new functionality. Okay. Yeah, so the protein folding problem. So, you know, it's a 50-year-old problem. And as you all know, for those of you who are actually in wet lab, you know, it takes months, maybe even years. It's a real challenge to experimentally resolve structure. And so I guess some of the kind of two principles that have been, you know, occupied the structure sphere, is that, you know, since structure can determine, sequence can determine the structure, as Anfinson's dogma stated, it could, in theory, as a kind of computational problem, be used to predict a structure. And then I quite like also as well the Leventhal's paradox, in the sense that, you know, if you were to go through every possible conformation that you can get from a single amino acid sequence, it's going to take an eternity. So it's a paradox in itself, because it takes you know, less than a moment to fold a structure. So that posed as an interesting challenge for the community for the last 50 years. And I think it's a really important figure to kind of remind ourselves as well. You know, the UniProt database has around 250 million protein sequences. And in comparison, the PDBE has just over 200,000 experimental structures, of which 60 are unique proteins. So you can see this gap is massive between the two, and obviously using predicted structure models was kind of is the solution to kind of close this gap. Okay, so AlphaFold came along, and as Arne has said, so it's developed by Google DeepMind, and it was shown an exceptional level of accuracy in the Casp 14 back in 2020, and this was kind of a real kind of showstopper in the sense that now we can predict with very high accuracy these predicted models. I was going to go through this, but I think I won't spend too much time on this, so I think I'll skip over this for now. But I kind of want to focus more on what you can do downstream with the models that we get from our AlphaFold database resource. So I thought I'll just highlight again as a reminder some of the strengths and limitations of AlphaFold, the things it can and can't do. The great benefit is it's using amino acid sequence, you can predict a structure model. And what's great about AlphaFold now, let's change the shift in focus in the community, is that it's really a great way as like a starting point to start exploring testable hypotheses, as Casper has shown you with AlphaFold what he's doing. And so there's other many more use cases where you can start exploring ideas very quickly. As you know, with AlphaFold models, they often use templates from the PDBE as a kind of, as a template, if the MSA is quite shallow. So this is kind of another, what I was meant to say, it's starting model for X-ray crystallography and cryo-EM. But yeah, so the templates as well. And most importantly, AlphaFold produces 3 outputs. One of them, obviously, are the atomic coordinates of the model that you're interested in. And there are 2 confidence metrics that you should use always together to assess the quality of your model. I thought I'd cover some of the limitations of AlphaFold, the things it can and can't do. It's a very extensive list, but I'll cover some of them. So it only accepts 20 standard amino acids. It can predict 5 models per run as the kind of default parameter, but often they're generally very similar, these models. So in other words, it is unlikely to predict conformational variability from a single run. It cannot predict assembly, so that's why we have AlphaFold Multimer. And we discourage you from predicting point mutations with AlphaFold because it wasn't designed for that. You also have, it was also not designed to predict the binding of ligand molecules or any post-translational modifications. And obviously we heard a little bit about transmembrane proteins. It's not aware of the membrane plane either. So if it doesn't look right, the model, it's probably because it isn't right in terms of where the protein starts folding inside the membrane plane. And as you've heard, if the protein you're interested in is an orphan protein, the performance will go down. Obviously, there's fewer evolutionarily homologous sequences. So as a result, MSA is shallow, and that can suffer, the quality suffers as a result. So these are just kind of some of the limitations that I wanted to highlight. Okay, so the metrics. For those of you who don't know already, there is the first PLDDT, so it's a per residue accuracy. And so what PLDDT does really is it just assesses the quality of each residue in this space. So we've color-coded the model confidence based on these 4 colors. So I'm just thinking. Okay, sorry, I thought I had a pointer. So we color code residues in 4 categories, so blue being confident and red, yellow being kind of poorly predicted. So it's not very confident. And then the next equally important confidence metric is the PAE. So what the PAE is predicted aligned error, and so that is for each data point there is a value for every residue pair in the structure. And in doing so, it really measures the confidence of the relative position for any given amino acid for your given model. And by looking at and to analyze the data for the PAE, you can use this, it's visualized as a matrix. And so as you can see, this I thought this is a great video to kind of highlight. So what's happening here is we're selecting domains along the diagonal to kind of highlight how they are in structure, because this is interactive and you can do this on our pages. But if you want to understand whether the actual relative position of any two given domains are confident, you would have to select the region that's shared between these two domains. And as you can see in a second, right now I'm selecting the region between these two domains. And since it's white, so white being not confident, green being confident, I wouldn't trust the relative position of these two domains. And so this is how you would analyze your model. Okay, so I think in the more interesting parts of this talk. So it's all great now that we have all these hundreds of millions of structure. But the question is, how do we now navigate? We have this treasure trove of data. So what do we do? So to give a bit of background, it's kind of important to remind ourselves, is that for over a decade, sequence-based search methods have always been dominant because we've had more sequences than structures naturally. And so when you want to infer function from a particular protein that you're not familiar with, or it's understudied, let's say, often you would use, you'd build a multiple sequence alignment of domains that has a structure you probably know the function of, and then you would build a sequence profile, and then with that sequence profile you'd search and apply the entire proteomes, whatever you're interested in, and that way you can start labeling certain protein coding genes for what function they may have. So that's always one of the main ways to identify the function of your protein. Now, because we had so many hundreds of sequences, the methods have always been optimized for speed. And so, comparison to structure-based methods, they're very accurate. There's a few of them like DALI and TM-Align, but they weren't initially designed to scale, gracefully, because we didn't have such large databases for structures. So we had this kind of, let's say, bottleneck in that sense. Now that we have this data, what do we do? Well, there is this fantastic tool called FoldSeek, if you haven't heard of it yet. This tool, FoldSeek, it was developed by the Steineger Group, based in South Korea. And quite simply, it enables you very fast and sensitive comparisons for large structure sets. And it has the level of accuracy and also the speed that we need. And quite simply, all you need to know about how this tool works is it essentially translates your 3D structure of interest, your query that you want to search with, into how they've defined a 3D interactions alphabet, and essentially pipe that into their own sequence-based search tool, which is MM62. It's also developed by them. And because sequence-based tools are optimized for speed, it's fantastic, and it kind of and it's fast for that particular reason. So for those of you who don't know FoldSeek, I thought maybe it would be nice to kind of show you why it's such an incredible tool for the community today. So I thought I'd focus on this one use case. I found this paper. So what they did with FoldSeek was they were interested in studying bacterial protein toxins. And so there are an incredible amount of bacterial genomes out there that we don't know in the environment. And so often there are these gene islands which have a combination of toxin, antitoxin, and various other proteins which are involved with virulence or abortive phage infection. So in essence, how it communicates and interacts and attacks its neighboring environment with other prokaryotic cells or eukaryotic cells. So as you heard from Casper about those toxins, that enterotoxins, right? So in a similar manner, so what they did with FoldSeek was they said, right, let's identify these hypothetical proteins in genomes that are understudied. So specifically, they looked at the Thaura phenylacetica. Sorry, probably butchered the pronunciation of that. And so what they did was they took these hypothetical proteins, and because we had models of them in the AlphaFold DB, they took these models and they searched with FoldSeek against AlphaFold DB and the PDBE. And what they discovered, you can see the figure in yellow are actually are known toxin and antitoxin proteins that have been characterized experimentally. Whereas the ones in blue are these hypothetical proteins in these predicted gene islands to be involved with the toxin and antitoxin relationship. And so I think if you just spend like a moment just to think, if you can do this with one gene island, imagine if you scale up to hundreds of genomes. And so hopefully this kind of gives you a little taster of the broader implications of what you can do with fold-seq. And so this here, they built actually a database here. You can read about it. So that's like on many hundreds of proteins that they looked at. And so yes, so it's a great tool. So we are actively collaborating with the Steininger group. And really the sole purpose of our collaboration is to enhance the depth and utility of the database. Okay, so the prediction pages. We will go through from kind of top to bottom what is already there and some of the things that we're developing now. You've probably seen this already. So this is the main homepage for AlphaFold DB. And so here you would search with your protein of interest or UniProt accession that you want to explore. And this is kind of one particular entry page that you would look at. So obviously you can see some metadata from UniProt about the protein name, the gene, and its description, and whether there are any experimentally determined structures for this particular UniProt accession. And then, of course, you have the 3D mole star viewer to look at your predicted model and the PAE plot. Okay, so as I've shown you before this example where you can look at hundreds of proteins, right? But I think before we started looking at fold-seq, essentially it was just a landing, it was just an entry page where you can assess your model. But that's it. If you wanted to do something else with it, you'd have to download it and do your own analysis. So what we wanted to do is, because these structures are kind of naked, apart from the metrics, they're kind of naked, and you need to add information to it. Without any biological context, you really can't understand these proteins. So we were hoping that with fold-seq, the easiest thing you can do is just compare it with homologous proteins that are better studied to understand, well, this protein of interest that you're looking at, what else can I learn about it? So for the rest of the talk, I'm going to kind of look at varying levels of granularity, let's say. So I'm going to look at very similar proteins, an arbitrary measure, to kind of moderately similar and distantly similar. So I'm going to focus on homologous proteins. So when I say homologous proteins, in essence, I am trying to find a group of proteins that share something in common. And in this case, it's sequence and structure. So for those of you who are familiar, we kind of, in collaboration with the Steininger group, used the two-step clustering workflow. So this is where they clustered the entire AFDB. And we display this information in two separate ways. And I'm going to walk you through first the method and how we display the data. OK, so let's focus on the first part. So what the Steininger group did is they took 214 million proteins from the database, and they applied the MM62 tool. So in other words, they're clustering purely on sequence. There's no structures involved yet. And so the clustering criteria that they used is as long as it's a minimum of 50% sequence identity and 90% sequence overlap. And in doing so, you create now 52.3 million clusters. And so how do we display this information? So if you haven't seen already our entry pages, at the bottom, you will see there's now a new table. So it's called Structure Similarity Cluster. And so this data that I explained to you in the first clustering step, where we cluster only in sequence, so that's displayed as this table. And then you can see it's this tab, and it's called AFDB50 MM62. And then I'm just going to also explain the second part, and then I'll walk you through bit by bit. So then from each cluster, what's done next is we take a single protein from each cluster, which has the highest average PLDDT. And then now we used fold-seq to cluster. And so here, you start clustering based on 90% structural overlap and a threshold of less than 0.01 e-value. And so then we're left with 18.8 million clusters. And so now this data is visualized in this other tab called AFDB fold-seq. So now we're viewing homologous proteins on these two clustering steps, one on sequence and one on structure. So let me kind of walk you through what you can do with this table. So, as I showed you, you can navigate between these two tabs to look at different levels of homology. So every row that you see in the table is a single entry. And you can see, obviously, the AFDB accession, the description of it, the species associated with it, and the sequence length and average PLDDT. And what you can do in terms of functionality with this table is you can, you can sort the members of this cluster by sequence length or by average PLDDT. And additionally, if you want to filter your species, you can use the taxonomic filter on the top right to kind of filter your table so you're looking at proteins only in a specific organism. Right. It's all great that we have this table there, but probably the question is, what can I do? What can I use it for? And what's the point of this table? So I thought I'd highlight some use cases of the things you can do. And maybe it might benefit your work if you haven't done already. So one of the things you can do, which is from an evolutionary perspective, is really to explore in this structure space. I think the key message is, how do you navigate the structure landscape? Is for your given protein of interest, across these homologous proteins, where is it expressed across the tree of life? Is it only bound in plants? Or maybe it's prevalent across the tree of life? Who knows? So by doing this, you get to understand a really good feel for this particular protein fold. Where does it sit in this structure space? Another, and I think the most powerful use case, I think I've kind of hinted on it, is to make educated guesses about the function of unknown proteins. So if you're looking at a protein from whatever study and there's barely any information, I highly recommend you check out the table, because very quickly you can find another protein which has certain criteria that matches it, and actually says, you know what, based on the function of this other protein, it's likely going to have the same function. So if you look at this example I've highlighted here, the protein in pink, I was kind of, I found it looking around, and there was absolutely no information on it. It's an unknown protein, and you can see the name of the protein is very uninformative. So what I did was I looked at the cluster table, and I found human, a homologous protein from human and from bacteria. And you can already see, based on the name, that it's possibly a kinase. So very quickly I learned about my protein of interest. Another very interesting thing, the use case with the table, is in gaining some structural insights about the differences or similarities. So I know it's a bit hard to look, but there's on the left side of this structure superposition, there is a missing chunk from the protein from Drosophila, the pink one. And because it's a kinase, there's actually a cavity where its active site is. But that chunk is missing, and the cavity is much bigger. So, potentially, maybe this function, the protein might be similar to these other proteins, but it might have lost its function. I don't know. So hopefully, by exploring the cluster tables, it's going to open up more questions about your protein of interest. And the reason why I was able to identify this missing chunk was because I just sorted by the sequence length. So you'll be surprised how many proteins there are which are missing maybe 100, 200 amino acids. And it's examples like this where they just kind of disappear. And so it's interesting to investigate that. And then on a more technical level, if you're not too bound by your protein of interest, you can just sort by PLDDT and find a better predicted structure model for whatever it is that you'd like to do. And just to kind of bring the message home, so I thought I'd walk you through a random protein that I found. So you can see there is absolutely very little information, and the name is not very useful. So let's say but at least the PLDDT and PAE is great. So at least that's the good thing that's going for it. So what I did was I explored the cluster table, and you can see there's about 5,000 entries that are homologous with this protein. So what I did was I thought I'd save myself time and start searching for species that have, as part of the reference proteome, so any model organisms. And long story short, I identified one protein from yeast. And as you can see, it's got the badge that it's reviewed. It's from a reference proteome. And conveniently, when you read the biological function, it's a delta fatty acid desaturase, whatever that means. I'm not an expert in this particular protein. But the point is, very quickly, I just learned about my protein, which was just before bare, and there was no information on it. So this is just something I wanted to highlight. OK, but let's say you looked at this cluster table, both on the sequence level and the structure level, and you still can't learn about your information. You think, right, is this a dead end? well, no need to worry. So one of the newest features that we're now currently working on, we're hopefully going to release soon, is we're going to let you, as the user, search with FoldSeek for remote homology of distantly related proteins. So this is just like a preview. I know it looks identical, this table, but this is a separate table unrelated to the previous one. So what you'd be able to do is on your entry page, you'd say, submit a job, and you'll be able to search with FoldSeek, and it's an e-value that's also relaxed, the threshold, so you can start identifying slightly more distant proteins, related proteins. And you'll be able to do as well two things. By searching, you'll be able to search against AFDB, obviously, but then a very important part as well is the PDBE. By superposing with experimentally determined structures, there's a lot of value in interpreting your model. So if you look at the table, it's mostly the same. It's a very similar functionality to the other table I showed you. But it's got additional sorting capabilities. So as this kind of metrics from FoldSeek, you can sort by sequence identity and also the e-value to kind of score the hit. And I thought I'd show you as well. So if you do a drop-down list, you can actually see the pairwise alignment of the two and the structure that it matches with. OK, so I throw this out there just to kind of emphasize my point. So this is pairwise alignment of these two sequences, both AlphaFold models. And you've got the conservation. It's a valuable conservation score. So I thought I'd ask you the question, are these evolutionarily related? And after looking at it for a while, you're probably realizing you cannot find a single column where the amino acids are identical. And just to give you an idea, it's 5.6 sequence identity, which is, if you looked at this, you'd say, right, they are obviously no way they are related. But I'm sorry to disappoint. It's actually this particular alignment. So what you see here is actually this particular domain. I searched with FoldSeek, right? And identified this particular domain. So what I did was I took this particular, yet again, another uncharacterized protein. And what I did is I searched on Interpro to understand if it had any sequence-based annotations. It was practically bare. There was no information. So by searching with FoldSeek, I was able to actually identify a particular domain in my uncharacterized protein against this other protein from chicken. So it's reviewed, and there is actually a functional sequence-based annotation for this particular domain. And I gained this from another resource, Interpro, and it's actually a DNA single-strand annealing protein. So just to also to emphasize my point, I went to the very bottom of the FoldSeek list. So this is the lowest e-value you can get from this search. And yet still, I was able to gain some information. Of course, I'm not saying these are definitely identical, these domains, and they have the same preserved function. You would need to investigate further. But the point is, I'm hoping to show you that I can, with these new features, we can equip you to start answering more interesting questions about your protein and start exploring really where your protein sits in this structure space, since there's hundreds of millions of these structures. And other use cases you can do. So again, when sequence homology is low, you can identify proteins. Potentially, you can also uncover analogous proteins, so by convergent evolution, so if you really relax the threshold, but that should be explored with caution. And as I think I hinted already, the ability to understand the diversity and function of protein structures makes this a really powerful tool to understand where your protein sits in the structure space. And as you can see, there are these two other domains that don't align in this structure. So I've just learned about this other protein that shares this domain. So there's another. It matches with this other multi-domain protein that has some other function. So I've learned a lot by just doing one search. Okay, I'm not a fan of this slide. For the last 20 minutes, I've been telling you how amazing structures are, but I think it's important to kind of bring ourselves back down to earth. So some of the limitations of structure-based search, and I noticed this with FoldSeek myself, it's less about the tool and more about how you use it in terms of the limitations. So if you're looking at a protein that is poorly predicted or has, let's say, low PLDDT in general, or in fact, you have a very well predicted domain, but there's tons of insertions in it, FoldSeek will struggle with it. And this is based on my experience using it. So if for whatever reason you're searching for a protein that has very little information and you hit a dead end, it's possibly not a dead end yet. You probably have to investigate further, maybe additionally look into isolating the domain. Another thing, another limitation is, again, I keep reminding to always use the PAE. So if you have, let's say, an active or an inactive protein which has a very large difference in RMSD with itself, maybe potentially that might affect the outcome of the results you get from FoldSeek, because you might look for only active or inactive. So I haven't personally experienced this. I don't know. I think this is just something common sense in terms of how FoldSeek works. And also, if you have two domains and you haven't looked at the PAE, and the PAE says, actually these two domains because the model shows that they're interacting, right? And you look at the PAE and it says, no, I'm not confident that these two are, the relative position of these two I'm not confident of, then probably the outcome of FoldSeek might not be the answer that you're looking for. So you probably should look at them as individual entities, these domains. So these are just some of the limitations, but they're very easy to fix. Okay, and so really, I just wanted to highlight what's kind of why I came here today is just a sobering reminder that there's only approximately 200,000 PDB entries in experimentally determined structures, and now we have 900 million predicted structures such as AlphaFold EBI and ESM Metagenomic Atlas, and in solving the protein folding problem, created a new challenge, which was how do you make sense of this massive data set, this treasure trove of structure models. So hopefully I've highlighted to you that, in fact, by clustering, grouping proteins based on sequence and structural properties, you can start identifying relationships between similar proteins and understanding how, across evolution, where it's prevalent. And finally, with the new live FoldSeek search, hopefully you'll be able to explore the kind of the function and diversity for a given protein structure that you're interested in, even when sequence identity is poor, as I showed you in the example. And before I close off, I thought I'd distract you with some pretty pictures. So, essentially at AlphaFold EBI, we're trying to continue empowering our users with new functionality and abilities to understand your protein very quickly just from the entry page alone. And so this is only as useful as it is to you. So if you have any particular use cases, or you see something that perhaps you think we should consider developing a particular feature, please either come talk to me today, or we've got the help desk, so just drop in a request and we'll try to get back to you. And on that note, I will leave it there. Thank you.
We have time for some questions. Yes, go ahead.
Yes, thank you for your nice talk. It's more philosophical question. You mentioned that there are 200 and so many million structures in the database, and even your even mentioned 900 million structures. I was just wondering how many structures are there on Earth that we can expect? What's the number what's the plateau?
I'd have to ask the bacteria and get back to you. I don't know. There's just so many organisms that are, in the environment, uncultured. I mean, I wouldn't be surprised if that was the tip of the iceberg, because there's just so many organisms out there. Not mammals, but I'm talking microorganisms that we just don't know of. Probably should put a number to it. Let's say, and then I'll revisit it. Yeah, I'll think about it, but I'll revisit it in 5 years. And
Thank you for your presentation. My question is more like a commentary and actually a little bit of a critique in a constructive manner. So I think one of the major drawbacks of the AlphaFold database is that in the area of membrane protein representation space, it actually provides nonsensical representations of the predicted structures in the following sense. For instance, in the case where you have a multi-topic membrane protein, which are actually many of them, where you have an extracellular component, a transmembrane region, and an intracellular component, the predicted structures are actually a convoluted representation of those domains in a nonsensical fashion, such that they crisscross each other. So for the trained structural biologists, that you can easily resolve that, because you look at it and you say that's nonsensical, and then I'll split them up and represent them in ways that make sense. But for the millions of others who are non-trained structural biologists and are trying to get into this, it's a very confusing exercise. So as a practicing, experienced structural biologist, I actually get comments from my colleagues who have some knowledge of the field, who are asking me to deconvolute those structures for them.
Mm-hmm.
So what is the EBI, EBI, EMBL doing to actually lower those thresholds and facilitate the implementation of the AlphaFold database among the communities that are non-specialists?
Right, so one of our main focus that we're actively doing, and hopefully over the next few months you'll start seeing some new developments from our team. So we are aware that there's this massive community who are all jumping now onto AlphaFold DB and misusing it, right? As you said, they might not have experience either in structural biology or specifically in a domain such as membrane proteins. So what we're trying to do in terms of mitigation, broadly speaking, we are now hoping to kind of unravel some sort of training materials to actually educate the community that are trying to use our resource, just to understand that how to use it and how not to use the models. Now, specifically for your use case that you explained, I think a very important reminder is that they are, after all, predicted models, right? And they should be always compared with experimental data if it is available.
Maybe I can explain my point well. I'm not questioning the accuracy of the predictions of the individual domains. I'm questioning the way they get represented in a manner that is completely nonsense from a biological point of view.
Well,
when you have the extracellular domain flipping out and being where it's where the cytosolic domain should be, and it's crisscrossing with the TM domains, and they're even at the same level, then actually, locally they are correctly predicted, but they're nonsensical from a biological point of view.
Yeah, no, I understand. I think all I can say is, right, AlphaFold it's not aware, like I said, the membrane plane, so it's not something we have control of yet. So
this information is out there. In Unicode, you have a delineation of domains according to their localization in the cell, extracellular, TM, intracellular. So that information could be used.
More work is needed.
Yeah, no, I understand. Yeah. So I mean, there's a lot. I mean, we can have a chat afterwards. Yeah.
More work is needed.
Yeah. Of course,
experts in the different fields and use actually the annotation that is already available to kind of untangle this.
And just one.
It's a huge amount of work. But
One comment, one of the features, I'll just end this on this, is that one of the next things I'm actually going to be working on is to actually map sequence and structure-based annotations on the models. So you'll actually be able to identify sequence-based profiles to say, oh, actually, extracellular domain XYZ. So hopefully that might mitigate it a bit, but yeah.
The first step, you have to plan them, and then you have to introduce
Yeah.
what is already available in other databases.
Exactly. So, yeah.
Yes.
I think, just coloring the extra and intracellular with the color is enough. A new view, let's say, a new layout. My question actually is about EBI's decision for representation of data in AFDB, or not representation. For example, the alignments are not shared that are used for model building. Why that is not shared? Is it a what was the rationale behind? And the other is for the forward seek, which I really love. If the final goal is to help, assign some biological function from other annotations, why don't you add the Unicode annotation score to the table, which would help users, pick out the one that likely results in some biological function? It's so easy to get from Unicode.
So the first question was MSA in terms of the actual prediction pipeline, right? You're not the first to ask. So I think it's one of those things that it's just a matter of like balancing between the cost of maintenance, because there's a cost incurring holding all these particular data for us to provide. So I think what we can...
Google has infinite amount of storage.
So, I mean, that's other projects take priority. Look, I very much agree with you. Yes, so the MSA is very valuable for those who want to use this, for sure. So all I can say is it's on our list of things that we are keep pushing with Google DeepMind, but we have other things that we have to do. So that's all. Right. Well, yes. So I mean, we have the metadata. So yeah, if you think that will improve, then yes, by all means, it makes sense for sure. So because we have all the metadata from Uniprot. So if there's anything that you think is valuable, then by all means, we'll definitely include it.
It all means that there's a lot of work for you to do.
Yeah, you are increasing my to-do list, just so you know. So.
Just a bit more maybe or scientific question. So you decide to do all your comparison on full-length proteins.
Mm-hmm.
And we know that domains are very important. And in principle, you have many ways to do them. You just much
Sure.
So have you why did you choose to do full length, not domains, or did you try the domains, or what would the difference be if you would?
Yeah. So no, that's a good point. So by doing full length, I think that there is already a lot of value in that, and that's the easiest thing you can do. There's nothing, you don't have to meddle with the actual structure or the sequence. So I think that's the first step forward. So you can very quickly learn about it. But I agree. So there were, as I mentioned, some of the limitations. Sometimes you can have large intrinsically disordered regions, which are these insertions, and that can interrupt the domain. So as I said, and I actually have a few examples if you want to learn more about it later, where I would search with FoldSeek and I thought, oh wow, there's only 6 hits. Surely not. Is this an orphan protein? Is that, maybe that was the dead end. But what I did was I actually started deleting some of these poorly predicted PLDDT. FoldSeek, thankfully, just stitches the breaks in the chains and thinks it's one long domain. That's what I did it. And I got nearly 1,000 hits very quickly. So yes, I do see the value, and I think as we were developing this, there were all these ideas, kind of like maybe we should possibly, build some sort of visualization to say, right, it has this domain and it shares with these other proteins.
So the easy thing would be to cut off, ignore all PLC less than 70.
For example, to put it very crudely, yeah.
That's probably easy way.
Many ways.
Excellent training material.
Thanks. I'll let my colleague know.
One more question?
Yeah, the last one.
Yes, please do.
All right. So one question from the people joined on the webinar. For more comprehensive structure function tools, not case by case, aren't we in need of a more uniform way of measuring and or storing functional data? Like ADMET score sheets in pharmaceutical industry.
Oh, wow. I don't know. I mean, can you repeat the question? I'm trying to think.
For more comprehensive structure function tools, aren't we in need of a more uniform way of measuring and or storing functional data?
Yes, I think in an ideal sense, in an ideal world, yes, it would be nice to have a one unified resource, gold standard of how to assess and transfer, let's say, functional information. All I can say is at EBI, since we're one of the leading bioinformatics institutes, I think we have all these fantastic databases for different resources. So in a way, we're under this umbrella, let's say, organization where I hope and I know personally from various experience where we actually collaborate with these databases. So in a way, we are kind of following kind of a, let's say, set of standards, but as a single unified, I don't know. It would be great, yes, but I don't know who would do that.
Linking to some of the other databases in a smart way.
Yeah, and that as well. Yeah.
Okay, thank you very much. Thank you for your talk, and thank you for the other speakers of this morning's session and for all the questions. This will help you do the work.
Perfect. Thank you. So I just leave this here?
Yeah, sure. Okay, so thanks again to our three speakers. I think we had a very interesting morning session, and thanks to all of you for questions. Also for the people from who joined online, thank you for participating, and thank you for your questions. So let's thank our three speakers one last time, and then hand over to the